Okay, very good morning. Very good morning. Good morning to esteemed guests, colleagues, and our loving delegates. So, greetings of the day. We welcome you all on behalf of Amity University, Haryana. It is an honor to stand before you today to deliver an opening remark for our Professional Development Program PDP on captivating theme of innovations, opportunities, challenges in the life science sector, skilling and research. So, this PDP is a testament to our ongoing memorandum of understanding with Life Science Skills Sector Development Council Government of India signed last year in May 2022. And it is a very befitting celebration of the World Youth Skill Day, which has just crossed on 15th of July 2023. The life science sector encompasses a vast range of disciplines from biotechnology to pharmaceuticals, genetics to computational biology, and the latest applications of AI in the life science sector, holding the platform to transform the very fabric of our society. This program aims to eliminate the cutting-edge innovations, vast opportunities, and crucial challenges that define this dynamic industry. So throughout this program, we will embark our journey to exploration, uncovering the advancements in life sciences that are revolutionizing our healthcare, agriculture, and environment sustainability. So I invite each and every one of you to join us on this enlightening voyage as we immerse ourselves in the realms of life sciences. So let us empower this for next 5 days by listening to the talks by the best scientists and the administrators from the industry. So let me welcome our director, sir, Professor Rajendra Prashant, who is a stalwart scientist. Let me giving him a brief introduction to deliver their welcome address today. So Professor Rajendra Prasad is an eminent scientist specializing in molecular biology, hold the esteemed positions from Director, Amity Institute of Biotechnology, Amity Institute of Integrative Sciences and Health, and Dean, Faculty, Science, Engineering and Technology. So with a remarkable career spanning of 40 years of Jawaharlal Nehru University, New Delhi, he served as a professor, Dean Life Sciences, Chairperson, Molecular Medicine, and esteemed Rector of the JNU. Having obtained his PhD from Center Drug Research Institute, Lucknow, and joined JNU in 1976, he further honored with his expertise in the postdoctoral trainings from the abroad university like University of South California, Los Angeles, and this career has led him to visit professorships of prestigious institutions like CNRS Paris, Catholic University, Technical University of Valencia, Bristol Myers Squibb in Princeton, University of Darmstadt, and New York Medical College. Professor Prasad has received numerous honors for his contribution to the field of molecular biology. Notably, he has awarded with the.
I think let us be brief.
Yes, sir. I'm briefing it with only three professorships and the awards.
It is joined. Good morning, sir.
Let me continue with his award of Professor Prasad, Royal Society Commonwealth Bursary, Alexander von Humboldt Fellowship, and Wellcome Founder Mercator Professorship at University of Bonn. So he is known to be an mycologist and as a member of all the international societies along with the national societies, including the Vice Presidentship of INSA. With the remarkable publication record from more than 300 research papers and 75 PhD theses and numerous number of the research projects for the government India and the international funding agencies. So Professor Rajendra Prasad's expertise, extensive research contribution and leadership positions have elevated him to the status of renowned scientist in the field of molecular biology. So let me welcome Professor Prasad for a welcome address today. Welcome, sir.
Thank you, my colleague, for a very elaborate introduction, which was not necessary. Our Vice Chancellor sir has joined. Good afternoon, good morning, sir. So this is a very important week-long activity which we have planned. And as our Honorable Vice Chancellor is also very ambitious plan for skilling sector. So it is in that line, we had planned this professional development program, especially on life science sector skilling. And in various sectors of life sciences, we have got numerous of experts who will be talking, giving their deliberations, and also focusing on skilling, upskilling in like infectious disease, nanotechnology, etc., etc. So it is a bit different from normal faculty development program. And actually, there was some situation. Our Yes. Gautam Bhattacharya ji, who is Chief Executive Officer of Life Science Sector Skill Development Council, had to rush to a ministry level meeting. So he would be now coming in the afternoon. But Dr. Himanshu from ISER Bhopal has very kindly agreed to swap his talk instead in the morning. So thank you, Himanshu, for this kind gesture.
Absolutely no problem.
So for this, your deliberations. And Mike, I will request on your behalf our Honorable Vice Chancellor to give his word of wisdom to kick off this important meeting today. Thank you, sir.
Thank you. Thank you so much, Dr. Prasad. Thank you for giving me this opportunity to connect to you, to extend a warm welcome to all our dignitaries who are sparing their highly valuable time to be with us at this very important faculty development program for life cycle sector skills, which are to be imparted both for the teachers as well as for the taught in universities and colleges around the world. I feel immensely delighted to be with you. I have a special word of welcome for Professor Kumar, coming all the way from ISER Bhopal, where I've also spent 6 years of my good life nurturing Rajiv Gandhi Technological University right from ground zero. And I was so happy that ISER has really been developed to great eminence in a very short period of time. And all this goes to demonstrate the kind of leadership, the commitment you have for nurturing science and scientific advancement in campuses and connecting it to the world of work, as well as creating that vital impact on the minds of people all around. We are so happy to have you. We would be equally delighted to have Dr. Gautam Bhattacharya in the afternoon, and all esteemed speakers who have been roped in by Professor Rajendra Prasad. Ever since Dr. Rajendra Prasad has joined Amity University, Haryana, we have been immensely excited to create not only facilities and programs, but also to engage seriously into research and innovations and creating capabilities and skills of the kind that the world would require in the life cycle, sciences sector. For people like me who worked in IIT, I saw Professor P.N. Srivastava redefining biological sciences and biotechnology as life sciences in the days when life science was not a buzzword in those days. People were saying, talk of biological sciences, talk of agriculture sciences, talk of medical sciences, talk of biotechnology, etc., or bioengineering. But Professor Srivastava said, I want to work in the area of life sciences. He raised very many people, including Professor Rajendra Prasad, and we are deeply benefited in his joining Amity University after spending years of his research-oriented professional career at JNU. During the last 7-8 years, we have created immense capabilities within our own campus here for life sciences research in areas which are important, areas like cancer research, infectious disease research, genomics, areas like biological computing, areas where we are looking at the large data analytics for understanding what's happening and how it is happening, including structural biology. But we are also looking at life sciences beyond medical sector, because we believe that the life science says that do not simply look at the medical science or design of the drug discovery or creating better understanding how we are inside as human beings and taking care of all kind of ailments and diseases that may come. It goes beyond. In my opinion, the life science sector covers 360 degrees of the human activities. It includes, in fact, not only just the agriculture sector or food sector, it also includes the environmental science sector, it includes the industrial sector. It certainly looks at industrial biotechnology as a very important area of activity. It looks into microbial kingdom in its bigger entity, as a bigger entity, and with a bigger impact it creates, not only on food and vegetation, but not only on human health, but also on overall environmental health and its connect. And therefore, I think in the life cycle sector skilling, we need to repeatedly echo words like integrative science, which means all sciences must work together in order to create the kind of capabilities of the kind that we require to trade and transact today with happiness, with cheers, with good health and with whole lot of work activity around, and continue to do this for good 100 years or more without falling prey to the diseases of the kind which we have encountered during the last 100 years of our scientific advancement. In fact, my mother lived for 101 years. She never visited any hospital. In fact, she used to tell me that you have been vice chancellor for long. Why not you educate people to remain happy and healthy without going to super specialty hospitals or consuming so many drugs and medicines during their lifetime. She used to tell me that, look, your food itself is a medicine, provided you know how to put it together, utilizing your life sciences sectorial skills and your lifestyle and the way you trade, transact, and behave also is important. The way you spend your time in maintaining your work environment as well as environment around, and commitment you exhibit for sustainability, for clean air, for clean water, and for ensuring the good health of the environment ultimately ensures that you don't have to go to hospitals. In fact, this is how, in fact, the human habitat on planet Mother Earth has survived for a good 10, 12, or 15 million years. In fact, during the last 100 years, we became so much submissive to the drugs and medicines and diagnostic tools and hospitals and super specialty hospitals now that we think that hospitals are going to create the good health for all, which is not going to be, in my opinion. And therefore, when we are confronted with great scientific advancement and technology innovations happening in all sectors of human activity, it would be important for us to conceive the layers of life sciences sectorial skills going far beyond medical biology, going far beyond looking at what biotechnology can do to confronting diseases, or for that reason, designing and discovering new drugs, etc. We need to, I think, take on board the advancement that we are making in all sciences and all engineering and put them all together and integrate, in fact, our life sciences sectorial skills for creating that happy, healthy, peaceful, harmonious humanity of which we wish to be part of. I thought this is an important agenda in Amity University. That is why we have set up an Institute of Integrative Science and Health. Of course, by health, we did not mean only human health. We meant environmental health. We meant health of everything that exists around, and therefore integrative science and integrative engineering, and together in the form of integrative science and engineering, are going to, in fact, take over the future course of actions and development that will happen from around. We in Amity University feel very happy that we are leading by example. We are not only talking tall, but we are also doing our work at the ground level and ensuring that the air quality in our campus is at least 3 times better than what it is in the National Capital Territory of Delhi. And also we are reprocessing every single drop of water, wastewater included, and we are reutilizing, in fact, the treated wastewater, asking for no water from outside to come to the campus for our needs. And this kind of leadership, by example, on all fronts, including energy efficiency and waste management, and also trying to see that we are managing our affairs with cheers and a smile, is important. And therefore, I think in the years to come, now the life science must graduate, in my opinion, to level 5.0, beyond what the world of automation, artificial intelligence, machine learning, big data analytics, and super specialties of medical could offer. I'm hopeful that during this program, many thoughts of this kind would be further deliberated and discussed from the eyes of the experts joining us from different institutions. And our teachers at the end of the day would be excited to see the bigger picture of life sciences, education, and research to be offered to creating that humanity of which we wish to be the part of. I wish to extend to you the warmest of the greetings on behalf of the Honorable Chancellor, Dr. Sim Chauhan of our university, and also on behalf of the Honorable Founder President, who had a vision, who has a commitment and a passion to see that science and technology is utilized to creating the compassionate, happy, healthy, and blissful humanity of which we wish to be part of. So thank you so much, Dr. Prasad, for giving me this opportunity to be with you right at the opening of this great dialogue on such an important topic that you deliberate and discuss how best 360-degree impact of life sciences, research and innovation could be created to create the world in which we wish to live with. Thank you so much indeed. Thank you.
Thank you, sir, for this word of wisdom. I know you are very busy and you're rushing to quite a few meetings, even at 12 o'clock going to Delhi. So I really appreciate that you spare some time
Yeah, I
for
will be jumping to listen to somebody else who is speaking, and another one where we are talking of artificial intelligence and augmented reality, and they are worried that human beings will become slaves of technology. I'm going to tell them that you cannot be enslaved. Vice chancellors cannot be enslaved. They have to remain on the top of the world, and therefore don't worry too much. Utilize technology for a good cause, and don't allow it to be utilized for a wrong cause. So, but thank you so much for giving me this early opportunity.
Thank you, sir. Thank you. Dr. Mack, we can proceed with the program.
Thank you, sir. And now, let me call Dr. Sangeeta to introduce our guest of the honor today, and Dr. Himanshu Kumar.
Good morning, sir. Good morning, everyone. Today, I'm happy to introduce Professor Himanshu. He is one of the finest scientists in the field of viral immunology and infectious disease biology. He did his PhD from Osaka University, Japan, in the subject immunology under the supervision of Professor Shiju as a Max Fellow, and postdoctoral fellow from the world premier International Research Center, Japan. Then he joined ICER Bhopal as assistant professor in 2010. He awarded various other fellowship like a Ramanujam Fellow, DST, Shimoto Foundation Fellowship, awarded as a Japan scientist for the promotion of science and technology, and many more on his accolades. He is profoundly influenced international science in the virology field, including the key contribution in the immunology and the virology. Now I would like to invite Professor Himanshu to deliver his lecture on the flipping of very interesting topic, flipping of the flu by the natural immunity. As old saying that you feed a cold and starve a fever. This is an old age and ancient wisdom that is further that is explained by the Michel Figueroa, how the internal innate immunology affects to maintain the healthy lifestyle from the various disease like a viral or a bacterial disease. So please welcome Professor Himanshu for the talk. Yeah, Professor Himanshu, yes, you can start your talk.
Yeah, thank you very much for, again, a very elaborative introduction. Yeah, I just do my thing, and you have given a quite elaborated introduction. It's not needed. Yeah, and now...
Himanshu, sir, it is required for at least for youngsters.
Thank you very much, sir. Thank you.
Thank you, sir.
So first, I would like to thank the organizer, Professor Rajendra Prasad and Dr. Sangeeta Kumari, for giving this great opportunity to interact with great people at Amity University. And I never been to the Amity University, so at least I can connect with all these great people. And especially I like the vision of the Vice Chancellor. He said that, okay, his mother lived for 101 years. So that is really very interesting. And believe me, sir, I still I see some people and who is living like that, and I always ask what you eat, what is your practice, what are all those things you do so that you are so healthy. Just for your information, my father is also 81-year-old, and he is a very healthy. He doesn't have any metabolic diseases, no problem, absolutely healthy. So this is a I think more about what we are taking and how we are taking the food, work, stress, and all those things. So if we learn these things from these great people who are living at our home, then I think we need not to go to any super specialty and all those things. So let me begin my talk, and now I will share my screen. Can you see my screen? Can you see my screen?
No, not yet.
So I'm clicking. So this is saying that host disabled participant screen sharing. So please enable me. Still it is flashing the same message, host disabled participant screen sharing.
IT, please do it so he can share it. Enable him.
Yes, sir. Please share now.
Okay. Now can you see my screen?
Not yet.
Yes, we can see it.
Madhukar,
Madhukar,
sir, Mac,
Where? Shall I start now?
Himanshu, 1 second.
Yeah.
Our Honorable Pro Vice Chancellor just joined. So we can take advantage of his words of wisdom before you start, if you don't mind.
Okay, no problem. So shall I stop sharing?
Yeah, I think for a minute, and then
Okay,
we'll invite.
sure. I stopped.
In fact, natural immunity holds the key for living 100 years without going to hospital.
Yeah,
We need to learn
that's
it
what I
more
am
from
telling,
that.
sir. My father is also very healthy. He's a 81-year-old, and he's no problem.
So,
And the word natural must be underlined repeatedly.
Yeah.
Not artificial, but natural immunity.
So is it clear? Clearly visible?
yes. Perfect.
Yes.
Okay. So the title of my talk is Flipping of Flu by Natural Immunity. So when I say flu, the slide is not moving. Can you see the change in the slide?
No.
I don't know. It is.
Oh. You close it and reshare again.
Okay.
Maybe.
Yes, sir.
Can you see the slide?
Yeah. Now try to change.
Yeah, it is coming now, right?
No, we don't see the change.
Okay. Can you see a red line below flu?
Yes.
Yeah, then
Yes,
it is
yes,
working.
yes.
Yeah. It's
Okay.
working.
Okay.
Okay.
Yes, now it is fine.
Yeah. So let's begin with this thing. So the title of my talk is Flipping of Flu by Natural Immunity. So when I say natural immunity, it means there are so many things. And first, I will give you quite great detail about the flu, the notion about the flu or cold. And then I will talk about the pandemic caused by the flu. If you, so during this pandemic, this SARS-CoV-2 pandemic, you probably heard the news that 100 years back, there was a massive pandemic. So I will talk about a little bit about that pandemic.
Will take advantage of you in the afternoon because then the chief guest is coming. Huh?
Shall I stop?
So are you in the room?
RP sir. Hello.
Shall I stop?
No, Dr.
Okay.
Himanshu, you can carry on, sir.
You continue.
Okay. So then I will talk about the disease course then host range and immune responses, and virus
Suno.
structure and challenges
Usko bhejo.
associated with virus infection.
Oh,
So, let's begin with. So first, I would like to explain what is flu and cold. So basically, when we say flu and cold, it means there is some set of symptoms associated with flu and cold, and that includes fever, headache, tiredness, body pain, and running and stuffy nose. There is a coughing and sneezing, sore throat, and all these symptoms basically makes it flu or common cold. This is what I want to say, that this is a set of symptom, and it is mainly caused by most of cases it is caused by rhinovirus, and now we know that this is also caused by coronavirus and respiratory syncytial virus, and this is also caused by influenza virus. So what is this influenza? Influenza word basically came from Italian word meaning influence, and this flu word basically came from this influence. So this is a influenza, means this is the literal meaning of influenza, and it is derived from the Italian word. And this flu basically caused the massive death about 100 years back in 1918. So we work on influenza virus, and this is one of most severe pandemic. Now we have seen the SARS-CoV-2 pandemic. So I can say that this is the one of most severe pandemic. And this pandemic, we call it as a Spanish flu. And this was started around 1918 to 1919, and it was there for several years. Like right now, we are still experiencing this SARS-CoV-2 infection here and there around the world. So it was also there. The Spanish flu was also there for several years, and it infected 1/3 of the world population, about 500 million people. And this caused massive death, about 50 to 100 million, or in Indian counting system, it will be a 5 to 10 crore. And in India itself, only 1 to 2 crore people lost their life during this pandemic. But when you find out the literature, then you will not get too much information about the India. But I was fortunate that I got one very nice video about this thing. I'm not going to show that video, but I have that video. If somebody is interested, I can show it to them. And since this virus caused a massive death, so there is some very means intriguing question in the scientist or virologist or immunologist, why this virus caused so much death. Okay, so here I will show you that this is a kind of motivation to find out why this virus caused the death. So to unlock the deadly secret of this 1918 flu, here I will talk about one very devoted scientist. His name is John Wilton. He wanted to understand that why there is so much death. Okay, so at the age of 25, he visited the Alaska, where the people who are infected with influenza virus, they died and they were kind of buried. And you know that this is a very cold place. So he thought that, okay, he can drive those dead body, and then he can isolate the virus. So here you can see that at the age of 25 year, he visited this place, Alaska, excavated, and then he tried to isolate the virus. Here you can see his, I wrote it, the Hilton's fatal passion. So he know that this is the most dangerous virus, and he is preparing solution, or preparing the virus by aspirating through his mouth. He could be infected if the virus is live. Okay. But unfortunately, at that time, those molecular biology techniques and all those things are not well developed. And unfortunately, he could not recover the virus. Then still, he visited at the age of 72, after again 46 years. At that time, these molecular biology techniques were quite well developed. But Again, he was not successful. So here, the aim of showing all this thing, that his persistent passion for the science, or he wanted to know, why this flu caused so much death. So that was his question, and he was quite persistent throughout his life. But after some time, this scientist Tampey, he basically isolated the virus from this formalin fixed lung autopsy, and this started the work of reverse genetics. So reverse genetic is a system by which you can isolate the nucleic acid, and then you can make the RNA molecule, and from RNA you can make a DNA, and then you can make a plasmid, and then if you introduce this plasmid inside the cell, then you can derive the virus. So this is a very complex system. It's not so easy, and the way I have explained, it has a lot of complication, because this virus is a negative sense RNA virus, and that has several layers of complexity. So end of the nutshell, in nutshell, I want to say that they finally recovered the virus, and then they investigated why it is so virulent. So they came up with these points. This virus is basically derived from avian virus. This rapidly infects the respiratory tract, and then over there, this will reproduce and produce very high number of progeny, and they severely damage the lung. And for all this thing, the viral polymerase, and there is a one gene known as HA. Later on, you will understand HA stand for hemagglutinin gene. So they play a most important role in lethality and causing the death. So when this work was completed, then there was one very big public controversy whether this work should be published or not, because if this tool or this virus will go in wrong hand, then that can be used as a potential biochemical weapon. But there is another school of thought that, okay, if it will be released to the society or to the scientific community, then there will be a better preparedness for such kind of virus if there is some pandemic in future. So after all these debate, this work was published in Science and in 2005. And this work is a really great work for this 1918 flu virus or pandemic, which caused the pandemic. Okay. So here, my aim is to say that, okay, this is very important. The influenza virus research is very much important. Now I will talk about how this flu virus infect. Basically, it infects the individual through mouth or nose, and generally when individual infected, then at that time they will not know whether there is a infection or not. But around day 2 and 4, there will be some initial symptoms, and this virus will settle in the lung, and at that time the individual will be highly contagious. They can infect. They will not know, or they will have a mild symptom, but they can also infect the other individual. They will not have a so severe symptom. Around day 5 or 6, the severe symptom will appear. There will be a fever, headache, muscle pain, which we also call it as a myalgia, sore throat, cough, and fatigue. So this is a kind of symptom which I have explained you in a beginning. So this is kind of common symptom which is associated with flu. Of course, there is a very high fever in case of influenza and other things, but it is very difficult to distinguish at this level. So once these symptoms has appeared, then there will be a ceasing of, or kind of a dampening of host immunity. And when this host immunity damps, at that time there will be chances of secondary infection, secondary mainly bacterial infection. And if the individual is immunocompromised, there is a possibility of fungal infection. So these bacterial and fungal infection basically cause or make the situation worse, and the individual is highly vulnerable. And if the individual overcomes, so generally in order to kick off the adaptive immunity, which is mainly mediated through antibody or T cells, this development of this immunity takes about 10 to 14 or 20 days. So if the immunity is kicked off, then the individual will feel better, and around day 10 and day 14, the individual will start recovering. But at that time also, the individual is capable to infect the another individual. They have a virus and they can infect the individuals. So this is a kind of, I thought to show this video, how this, here you can see the virus is infecting the lung cell, and this virus is released. And then it is, this is basically infecting whole lung and trachea. And when this is the situation, then they will start liberating virus and another individual can infect. And in that way, the pandemic can, this infection can spread, and then that will go to the epidemic, and then it will be, if it will cross the continents, then we can call it as a pandemic. Okay. So the pregnant women are highly susceptible to the influenza virus. People with chronic medical condition, now you are probably very well aware that there is several metabolic diseases or cardiovascular diseases. So individuals who are having these kind of condition, they are highly susceptible. The elderly people also are highly susceptible to this infection. And of course, the healthcare workers, because they handle the patient, they are also highly susceptible to infection. And above all, the children under the age of 5, they are highly susceptible. So, so this virus, the most interesting thing and the most dangerous thing about this virus, that this virus is present in almost all living animals. Okay, here you can see that this virus can infect the whale. Here you can see there is a whale, and this can infect the seals, birds, variety of animal, pig, cat, ferret, horses. And the most important reservoir of this virus is this wild aquatic bird, which is which harbors variety of this kind of viruses. Probably you might heard that there are so many kinds of influenza virus. I will come to that slide, and then you will understand why there are so many kinds of viruses. Okay, so many kinds of influenza virus. So basically, this is the serious problem. This is the source where through that this virus can jump into the human, and when they will jump into the human, then they will cause the severe disease. Okay. And the most important thing is that these animals, besides human, all these animals, they also have an immune response. But you can see in subsequent slide, the response is quite variable. Okay. Here you can see that this natural or natural reservoir, this is aquatic bird, they are basically asymptomatic. When there is an infection of influenza virus, they are having a almost asymptomatic, and they do not activate immune system so strongly. Okay. And since it is not inducing immune system so strongly, so there is a no so much severity. Here I just want to bring to your attention that so most of this, even influenza and SARS-CoV-2 infection, the problem is when this virus infects, it activates the immune system so much that there is a kind of situation which you probably heard, that there is a cytokine storm. Okay, and this cytokine storm basically causes the immunopathology. Okay, so here you can see that the natural reservoir, that is aquatic bird, they do not induce so strong immune response. There is a transmission host, and they also induce very mild or almost no severe response. But in terminal or spillover host like human and chicken, they react or they induce very strongly the immune response, and this is the point of problem. There is so much immune response, and this immune response start causing damaging our organ and organ system, and then that will cause to the fatality to the patient. So this is a point where you should understand this thing. So now I will talk about the influenza virus. So influenza virus here, I would like to say that this is a negative sense single-stranded RNA virus, and the genome of this virus is the segmented. Okay, there are 8 segments of RNA is present in the virus. Okay, and if you join each segment, then the total size will be about 13.6 kilobase. It's a not base pair. Remember, this is a RNA. Okay, and the virus is something. This is a schematic of this virus, and here you can see there are a variety of protein, and this virus is an enveloped virus. This envelope is basically acquired from the host. Okay, and there is a protein like a M2 protein. So M2 protein is basically essential for uncoating. They take the protons and then this virus will be uncoated inside the cell. Okay, there is a NA protein. NA stand for neuraminidase. Okay, so neuraminidase is very important for this virus for replication. So I will show you in a next slide. When this virus infect, then for release of this virus from the host cell, there is a need of this enzyme neuraminidase. Okay, so this neuraminidase enzyme cleaves the sialic acid, and then the virus will be released. And if you block this neuraminidase enzyme activity by some way, by some small molecule, then you can control the virus infection. Okay, another protein is a HA protein, which is also called as a hemagglutinin. So hemagglutinin is very much important for the binding with the host cell. So there is a molecule known as sialic acid, which is present on the host cell. So that sialic acid basically binds with the HA, and then it will initiate the infection. Okay. Another important point is here you can see that there is a polymerase complex. So polymerase complex is basically consists of 3 proteins, PB1, PB2, and PA. So this polymerase complex, please remember this protein PB1. PB1 is one of subunit of polymerase, because I'm going to use this PB1 word in my subsequent slide. So please remember this PB1 is one of subunit of this polymerase complex. And if you disable or mutate or do anything, kill the activity of this any of this subunit, then you can control the viral infection. Okay, so this virus is something like that. Here you can see the segmented genome is arranged. There is a matrix protein. So M1 is a matrix protein. Basically, it is providing the kind of framework to arrange this genome. Okay, and there is you can see there is a projection of this protein Na and Ha, and this is the whole structure. Okay. This is an electron micrograph, and you can see that this virus is looks like the way I have shown you in the previous animated schematic. Okay. So this virus is basically here. I'm showing you a very simplest form of how this virus replicates. Basically, it interacts with Na protein and this HA protein, and this HA protein is basically here you can see that this HA protein is interacting with sialic acid. Here you can see there is a sialic acid. Okay, and this is phagocytosed, and then there will be uncoating, and then there will be a release of genome. Then there is a transcription, translation, and synthesis of viral RNPs. And there will be a protein synthesis, because all these protein enzyme and all those things are need to be synthesized. And you know that virus basically hijack the host cellular machinery, and then they make their own copy. And then finally this virus will bud off. And this budding is very important in order to infect another cell. And this is basically mediated by the neuraminidase enzyme, which I have mentioned in the previous slide. And here you can just for simple understanding, here you can see that assembly of car on here there is a manufacturing of car. So all these things are something happen like this inside the cell when the virus infect the cells. Okay, and I told you there are several neuraminidase inhibitors, and most probably you may aware that this Tamiflu word. So Tamiflu oseltamivir. This is basically a drug which inhibits this neuraminidase, and then we can be protected against the influenza virus infection. There is another drugs here. You can see that this is a palmavirvir and zinamivir. All these drugs are some or other derivatives of this NA inhibitors. But now there is a one serious problem. The influenza virus developed the drug resistance. Now there is an emergence of this Tamiflu or this drug resistant influenza virus. So this is a very complicated situation. If these viruses are released or they will be successful in infecting the individual, then this will cause another severe pandemic. Okay, and it's difficult to control those infections. Now I will tell about the challenges and bottleneck with this infection and what we can do it. So there are 18 kind of hemagglutinin protein, and there is a 11 kind of this neuraminidase protein in the viruses, different viruses. And if you simply multiply these number, then you will get about approximately 200 serotypes. Okay, but this is just a very small number. As I told you that this virus can infect the aquatic birds. Here you can see that this aquatic bird, or this can infect another birds or another animal. And these animals are basically the factory for the generation of very dangerous viruses. Because how it is possible, for example, if any bird is infected with different kind of influenza virus, and now you understand that the genome is segmented. So there is a possibility that the different segments can be acquired from different virus, and that may result to the generation of very dangerous virus. Okay, and this is basically mediated by various processes, which we call it as an antigenic drift. Okay, which is basically a minor change. Here you can see that I tried to explain you with this simple example. There could be a single change. Here you can see that meat will change to the meat, P will change to the P. There could be two changes like piece to piece. There could be an insertion, high to high, hole to hole, and there could be a deletion, and that will cause that will again result to the generation of different kind of HA and NA, right? There could be a bigger changes. There could be a major changes like turnover to overturn. These are just simple example. And all these changes basically result to the multiple genetic reassortment. And that will result to this is something like that. There will be a change in sentence and change in meaning. And then there will be a generation of highly pathogenic influenza virus. So this is a very serious problem. Try to understand. Okay, still we in a year or a few years we see the pandemics. Okay, here I'm just showing again a very good animation. Here you can see that genetic reassortment. The cell is infected by different kinds of viruses, and these animals are basically a factory. Okay, and then they can make a different permutation combination of genome, and then this can generate the highly infectious influenza virus. Okay, so our current effort is against the flu is something like that. I just want to explain my frustration about the influenza virus. Here you can see that this animal, this whatever animal you can see, the animated animal, this guy is keep on changing, and our effort is something like that. We are putting a lot of effort, but still this effort is not sufficient to completely clear the or to overcome the influenza virus. Okay, so now I will talk about the first line of defense, because the reason is that once I will explain the first line of defense, then I will talk about our work, and our work basically given and hope that we can control all kinds of influenza virus. So let me introduce about the first line of defense. You know, the first line of defense is basically mediated by a variety of things like skin. There is a acid and all those things. I'm not going to take you in that detail, but our cells, all cells, basically immune cell as well as non-immune cells, they are capable to fight with any kind of infection. Why? Because of this. So our cell basically expresses a variety or broad variety of pattern recognition receptor. So here you can see we have a pattern recognition receptor on cell surface. We have a pattern recognition receptor in the endosome. We have a pattern recognition receptors in the cytoplasm. They can sense the pathogen origin molecule, and then all this sensing result to the activation of NF-kappa B. Probably you might heard that NF-kappa B plays a very important role in production of pro-inflammatory cytokine, and this can also activate a family of transcription factor, which we call it as an interferon regulatory factor, and this will induce the type 1 interferon and type 3 interferon. Okay, so please remember in this slide, just keep your focus on this molecule RIG-I. So what is RIG-I? RIG-I is a cytosolic RNA sensor, and this sensor distinguish between our RNA and viral RNA. And once this viral RNA is sensed by this molecule known as RIG-I, it will activate this transcription factor, and eventually it will activate to induce the type 1 and type 3 interferon. I'm going to introduce what is type 1 and type 3 interferon, and it will also induce the pro-inflammatory cytokine. Okay, so just remember few molecules. Please remember one molecule known as PB1, which is one of subunit of viral polymerase. Another molecule is RIG-I. Basically, this RIG-I senses the RNA molecule, and it induces the effector response through the production of type 1 and type 3 interferon and pro-inflammatory cytokine. So basically, type 1, type 3 interferon and pro-inflammatory cytokines, they are basically antiviral in nature. Okay, and I will explain you how they develop the antiviral state in the cell. So first I will take about the type 1 and type 3 interferon. So basically, you know that viruses hijack the host cellular machinery, right? So in that scenario, you cannot kill the virus. The best way to avoid the virus is to kill the virally infected cells. So the type 1 interferon basically induce the apoptosis of virally infected cells. Okay. Another thing what the type 1 interferon can do is that they make the healthy cells, those cells which is not infected by the virus, they make those healthy cells resistant from the virus infection, as you can see in this cartoon, right? And another, the most important thing which can the type 1 interferon is that, okay, the cell is already hijacked by the virus. So now what it can do, it can depress the cellular machinery. Here you can see that when the cell is treated with type 1 interferon, the cellular machinery will be depressed. Okay, and this will initiate or prepare or activate the professional immune cell for the development of virus-specific immunity, which is basically mediated by the B cells through production of antibody and T cells, basically cytotoxic T cells, okay, in order to specifically kill the virally infected cells or block the viruses. Okay, so this is the role of type 1 and type 3 interferon. And the pro-inflammatory, how pro-inflammatory cytokine works. So pro-inflammatory cytokine is basically an alert system. So when there will be a production of pro-inflammatory cytokine, then this is a kind of alarm in the body. And this alarm is basically a request to the all professional immune cell. Here, I just depicted with this Spider-Man and all those things, the specialized people. So specialized cell will come at the site of infection and then clear any infection, including virus infection. Okay, so pro-inflammatory cytokine work like that. But there is a problem. If there is a too much production, then that will cause the immunopathology. Okay, I will show you in a short while. So here, this is the situation. So initially, when there is a virus infection, our immunity is kind of weak, and here virus is kind of overpowered. But as soon as the cell will start producing the type 1 interferon, okay, here you can see, when the cell will start producing the type 1 interferon, then immunity will be heightened up, and then this can easily manage the viral infection. And but this takes some time. Once the type 1 interferon will be kicked off, that will also activate the adaptive immunity for production of antibody and production of virus specific cytotoxic T cells, and then we will feel better. Okay, so in general, if you see very carefully, we cannot treat the viral infection. Generally, we don't use antiviral drugs. What we do when there is a virus infection, including corona and whatever, only in few cases we use the antiviral drug, which is very cytotoxic. Okay, so in general, what we do, we basically manage the symptom. If there is a fever, you will give the drug which can reduce the fever. If there is a hypotension, we will give the drug which can increase the blood pressure, something like that. We basically manage the virus infection. We don't clear the virus infection. We don't give antiviral drug in general. Okay. So you have seen all this thing, and all these things need to be very tightly regulated. Okay. So you can understand the importance of regulation by looking at this image. If there is no regulation in the traffic, then everywhere there will be a chaos. Okay. And if there is no regulation in our classes, then this will be also lead to the chaos. But if there is a perfect regulation, then things, then there will be a better performance. Okay. So here I'm going to introduce about the one molecular switch, which we call it as a microRNA. So I basically work with microRNA. Okay. Why? Because this is very interesting molecule. They are regulatory molecules, and they are basically produced in our body in order to control our gene. You understand this slide very well. You can understand there is a transcription, translation, and then protein synthesis. And we know now that there most of genome, only 2% genome is encoding the protein. And now we started understanding the other part of genome is playing very important role in regulation. And that part of genome also encodes some non-coding RNA, like long non-coding RNA, circular RNA, and microRNA. So I work with microRNA. Okay. So this microRNA is produced in the cell in order to control the transcript. Okay. And basically it will bind with 3' UTR, and basically there will be several things. This can be degraded. The bottom line is that the transcript amount will be reduced in the cell, and then the expression of protein will be reduced. And that will be, this protein could be a negative regulator, positive regulator, or anything, right? And in that way, this microRNA is basically a fine tuner. Okay. So I have one very nice video about this microRNA. I thought to share with you to understand. So I will play this video. I hope you have enjoyed this short video. Now I will talk about my own work and I will try to present you in a very simpler form because it's a quite complicated. So our aim is to find out, okay, we produce the microRNA and this microRNA also regulate the innate immune genes, okay, the signaling molecule as well as effector molecules. So my simple question is that can these microRNA interact with the viral genome or not? If it is interacting, so what will be the disease outcome? And if it is not interacting, what will be the disease outcome? So this was my simple basic question. And again, I would like to emphasize on this molecule. So this is very well known that this RIGA is a RNA sensor, a viral RNA sensor, and it induces the type 1 interferon and pro-inflammatory cytokine. So we begin this work with a lot of screening, and we have performed a lot of bioinformatic work, lot of experimental screening, and finally we came up with one microRNA which plays a very important role in regulating the influenza virus. So here you can see that this is a RNA virus which is infecting the cells, and this is, here you can see this, there is a viral RNA, and this viral RNA will be sensed by the RIGI, and this RIGI is basically this is a protein which is produced by this RIGI transcript, and upon sensing there will be a cascade of signaling, and there is a production of inflammatory cytokine and type 1 and type 3 interferon, and it will induce or develop the antiviral response, which I have explained you earlier. Okay, so we here this is a scientific data. Here you can see that we have performed a lot of screening, profiling, and all those things. And finally, we come up with these microRNA, microRNA 205, microRNA 485, microRNA 125A, and microRNA 146A. Okay, so we basically this is the screening outcome, and then we perform the experiment. And here you can see that this cell, this is human small airway epithelial cell, HSAEC. Okay, so this is a primary cell we have used, and this is a A549 cells, and A549 cell is basically lung carcinoma cells. Okay, we infected the cells with influenza, and not only influenza virus, we use variety of RNA viruses. Here you can see there is a NDV virus. Okay, so we found out that this microRNA is basically induced, microRNA 485. Okay, so now the simple question is how this microRNA is produced and how it is basically regulate the transcript of RIGI. Okay, so by simple analysis, we could find out that this the transcript of this RIGI has two sites for this microRNA. Here you can see. So we performed a simple luciferase experiment. I'm going to explain you in a simpler way. We performed the luciferase experiment, and then we found out that when we mutated this microRNA site, here you can see there is a recovery, and without mutation, there is a reduction of luciferase enzyme activity. Okay, we have confirmed these experiments by a variety of experiments. There is an ego pull down experiment and all those things. Okay, just now in previous slide, in video you have learned about the agonate protein. So agonate protein is one of very important protein which is making a component of RISC, RNA induced silencing complex, along with microRNA and transcript. Okay, so we performed the pull down experiment and we have shown it by various methods. So now we know that, okay, this RIGI is binding and it is degrading the RIGI transcript. Okay, now you understand that if RIGI amount will be less in the cell, and if I will introduce this virus or infect the cell with this virus, then there will be an increase in viral load. Okay, because of low expression of RIGI. If I overexpress this microRNA inside the cell. Okay, so we got a same result. Very interestingly, we got this same result. Okay, when we have used the another virus known as NDV, Newcastle disease virus. Basically, Newcastle disease virus, we use it as an experimental virus because we can easily work with this NDV. We don't need a specialized facility. Okay. So whatever phenomena we see, we first tested with this experimental virus, which cannot infect us. They basically infect the birds and cause some problem, but not so serious problem to the human. Okay, to the normal human. Okay. So we use this NDV and we perform this experiment. And here you can see in this schematic, since the Riga is less, the viral load will be more in the cell, as you can see in this figure. Okay, there is more viral load when I have introduced this microRNA. This is plasmid expressing this microRNA 485. Okay, so generally when we perform this kind of work, then we want to make this work much more interesting. So I thought, okay, let's use the influenza virus. Fortunately, we had a BSL-3 facility in the Bhopal at that time. So I have used the influenza virus, okay, and I got a very interesting result. So the most interesting result is at initial time point, I got the same phenomena. There is an increase in viral load, but when I incubated the cell for longer time, 24 hours, the viral load was significantly reduced. Okay. So I, of course, I banged on my student, you must be done something wrong, this, that. But we have repeated this experiment maybe 10 times because we are not able to understand what is the problem. Okay. Maybe cells are dying or something. We tested everything. So finally, we got this stable result, and then this lead us to think why it is happening. Okay, so I have analyzed all those experimental thing, and finally we learned that when there is an influenza virus infection, of course there will be a genome. The genome will be present in the cell. This will activate the RIG-I, induces microRNA 485, and it will also, this microRNA can also bind with the RIG-I transcript and reduce the expression. Okay, so far so good. But why the viral load is reducing when I'm incubating with longer time period, as well as when I'm infecting, when I'm infecting the cells with higher doses of virus, the cell become a much more healthy. So that is a very interesting and very surprising thing. So we have analyzed the genome of this virus, and very interestingly, we found out that this microRNA can bind with the transcript of PB1 gene. You remember PB1 gene? PB1 gene is playing very important role in polymerization of nucleic acid of the influenza virus. This is the one of subunit of RNA polymerase, viral RNA polymerase. And here, if you see very carefully, this microRNA can bind with all kinds of influenza virus. The very old influenza, this is 1902, and here you can see that there is a 2014 virus. Okay, so this is a one of very big discovery in our group. Okay, and when we are introducing this microRNA inside the cell, then this is binding, and that makes the virus much more healthy. Means the cell become a much more healthy, and they are kind of not showing any cell death or cytotoxicity. So what is happening in this situation? So when this virus infects, then there will be a induction of this microRNA, and since this microRNA is in a particular amount, and you know that when virus hijack the cell, then most of cellular machinery synthesizes this viral genome, and the viral genome is in very high number in the cell. Okay, and in that scenario, since this is a constant amount, the most of microRNA start binding with the genome of influenza virus. Why? Because of more concentration, as well as the affinity of this microRNA to the genome is much more higher compared to the RIG-I. So in that way, this microRNA is basically acting as a double-edged sword. Okay, so double-edged sword means this will free the RIG-I, and this microRNA will bind more to the genome. So this will be basically degraded, and in that way there will be a this microRNA will basically act as a double-edged sword. So we have shown so when we demonstrated this thing, then reviewer had a lot of challenging questions. So what we have demonstrated this thing by using the reverse genetic of this influenza virus. Here you can see that the PB1, so PB1 we have mutated, okay, and we again demonstrated this, we reversed this phenomena when we have used this mutant influenza virus. We have used this system, okay. So in that way, there will be a sustained expression of RIG-I, and then this microRNA basically induces the anti-flu response by direct targeting the flu polymerase. So here you can see this molecule is synthesized by our body, okay, and this is basically targeting the influenza virus, okay. So this is one of very important discovery in our group. And I will show you one more work. Here I just want to say that we basically did this discovery by chance. It's a serendipitous discovery in my group. Okay, we basically wanted to improve the quality of work by using the influenza virus, but we came up with the novel this phenomenon. We performed all these experiments in biosafety level 3 laboratory. Here you can see this is a very expensive lab, which fortunately we had this kind of facility in the Bhopal at that time. Now I will talk about my another work, again in a very simpler form. We have discovered one microRNA 324, and this also restrict the influenza virus, and this microRNA also target the PB1. Okay, again I will show you in a schematic form. Here you understand when there will be a virus infection, there will be a more viral genome inside the cell, and this viral genome basically there is a one microRNA 324, and this microRNA again targets the PB1 of this viral genome. Okay, so basically this is constitutively expressing inside the cell. Okay, and this microRNA can also so when this will target the PB1, there will be a reduced viral load, and this microRNA can also bind with one of negative regulator of JAK-STAT pathway. So to understand the JAK-STAT pathway, this is a pathway which increases the activity, or it basically induces much more type 1 interferon if this microRNA will target the negative regulator. Okay, so this targets this negative regulator, but what this virus is doing when it is infecting the cell, it basically and of course it will reduce the viral load, and it basically reduces the expression of this microRNA 324, and basically this is a kind of immune evasion strategy. Okay, so this immune evasion strategy is used by this virus in order to establish the infection in the host. Okay, and here you can see that this virus is so smart. Here you can see this data. Okay, so this virus is so smart that they are making the changes in the genome. Okay, please remember the virus cannot make major changes in the polymerase. If it will make the major changes in polymerase, then identity of virus will be completely lost. This cannot polymerize and this cannot synthesize the thing. So here you can see that this virus is so smart that this is basically creating a mutation without changing the amino acid. Okay, here you can see these are the different influenza virus. Okay, and this is in order to evade the host immunity. Okay, so in that scenario, we can say that this, if we introduce this stabilized form of this microRNA, we can use it as an antiviral therapeutic. Okay, so this is another very important discovery in my group, and this is overall what I can say, that this microRNA 485 and microRNA 324, which can target the PB1, can be used as a therapeutic agent. But it is too early to say, because the reason is that when you introduce in human this microRNA, there will be so many targets. We need to take care of all those things. But yeah, they are one of good candidates, okay, which can be used for the treatment of influenza virus infection. And we need to perform all those human or in vivo experiments, okay. So whatever data I'm showing, it is a all in vitro experiment. I did not perform experiment in human or in mice. So before saying that this could be a potential drug, we need to assure that this will have a less side effects, okay? Because these microRNA are synthesized in order to control the host gene. So of course, we found out that this is targeting the influenza virus genome, but this is also very important to establish this thing, that there will be a less off target and all those things. So overall, I can say that this microRNA may be a key game changer in flu, okay? And this is our publication we have published in Science Signaling. This is little quite early we have published, and this we have published in Journal of Virology. And now I will stop here. I will thank my all these collaborators. Dr. Ashwin Raut, my previous mentor, he provided some plasmid for performing this antiviral studies. Akinori Takaoka, he is from Japan, and Ron Fauché. So his contribution is a very key. Ron Fauché basically provided the reverse genetic system by which I can create the influenza virus in the lab. I can mutate the virus and I can create the those viruses. I have shown you in my slide. And Peter Please, basically, he provided the NDV with GFP. So that is a one very important reagent in order to investigate the signaling pathway. Please remember, I did not show too much data because I thought it will be too much complex. And these are the funding agencies which supported this work. And these are my previous student. He is Sushil Kumar. Right now, he is a faculty at IIT Bombay, and all these guys are in University of Washington and different places in USA and UK. With this, I will stop here, and I think I can say that flickering of flu may be possible in future to avoid the flu pandemic. Thank you. Thank you very much.
Thank you so much, Professor Himanshu. A very insightful talk. And really, we know much of thing about the influenza virus. Hope our participant must be benefited by your talk. And once again, thank you so much for giving us a valuable time. Now we are open for the question answers. Shall we start?
We can start the question answers.
All right, right.
Compliment, sir.
All right.
We have
So,
few questions in the chat, so we can
yes,
put up these
yes.
questions.
First, I want to ask one very important question. Professor Himanshu, can you give insight on the flu vaccine and cross reactivity?
Yeah. So flu
It's a
vaccine
very important
is,
question.
Yeah. It's very important. So basically, flu virus, this vaccine which we people use, there is no guarantee. Okay, if this vaccine is used this year, this will give a protection to next year flu. The reason is that this HA and NA is keep on mutating. I showed you that there are different kinds of HA and there are different kinds of NA. And you probably remember that there is an antigenic shift and antigenic drift. So these mutation basically create a different kind of virus, okay? And this will not assure that, okay, the vaccine which you have used for last year virus will give the protection to this year. So there is a great variation, okay, because of variation in the virus. So the response will be also, there is no ensured response. We technically call it as an antigenic sin in the term of immunology. The virus is changed, the antibodies or T cells response are residing in the cell is against the previous version of the virus. So since the virus is changed, so these antibody and these T cells will not work. or there will be a less efficiency. Yeah.
Thank you. Thank you so much. Another question from Dr. Costa Bandyopadhyay. How
Yeah.
effective will be an influenza virus vaccine in India? In US, it is a rather a failure. Is it better to invest in discovering a drug targeting mRNA or polymerase than investing in developing a vaccine?
Yes. So vaccine. Okay. Let me give you the brief introduction about the vaccine. So when we make a vaccine, probably you are aware about this SARS-CoV-2 vaccine. Okay, there is a mRNA vaccine. There is a adenovirus vaccine. So over there, what we are using, we are encoding this protein, spike protein in the adenovirus, and then this adenovirus, when we introduce inside the human, then there will be a generation of antibody, right? So this is a basic concept, but please remember, again, it will be the same situation. I think we should not invest too much money in the creation of vaccine. Rather, we should because you understand, the face will keep on changing. Then how you will identify the enemy? Okay, so this is a really big challenge. That's why it is failure in US, and I am quite confident if we will put effort in India also, it will be not so successful. So rather we can target. So now we have to shift our focus. If we target the polymerase, the way I have explained you, we can give these two molecules, but I told you this needs a more deeper investigation in vivo condition in human. Okay, and that is not easy. Okay, so if we can use this kind of, if we change our target rather than surface, if we change the target to the polymerase, because viruses cannot afford to change the polymerase structure. If they will change, the virus cannot replicate, right? So I think putting more emphasis on these polymerases and all those things will be much more better and much more sustainable in terms of investment and all those things.
Okay, thank you. There is another question from Nelmar Saison. Does too much of protein in the body means we could easily be affected by any virus?
No, you cannot eat too much protein. This is a practically, I could not get eating too much protein or having more protein in the
body, having more protein in
no,
the
no.
body.
There is some steady state. You cannot just make more protein in the body. That is practically impossible. I don't know if there is any way. So whatever protein you will eat, there will be some steady state in the cell. The cell will not take more than that level of the required amount of protein, right? It will be thrown out.
Thank you. There is another question from Dr. Shalini. How is the seasonal influenza linked to the other types of influenza?
I told, yeah. So there is so the flu and cold is basically caused by a variety of things. As I told you in previous or in the beginning of my talk, this is mainly caused by the rhinoviruses. Okay, there are a huge family of rhinoviruses. So this caused the flu and cold, and sometimes it is caused by the SARS-CoV. Okay, you now you know that. So previously, coronavirus was not considered as a very dangerous virus, but after this this pandemic, now we recognize that this guy can also cause the fatality. Okay, so this can be also caused by the influenza virus. So in general, we are not infected by the influenza virus, because if there is an influenza virus infection, that will result to the severity. Okay, and in general, we are infected with the rhinovirus, and we think that this is an influenza virus, because the flu, yeah, there is this thing also, based on the genetic of the host, the flu can be also managed. Okay, not all individual will show the severity. Okay, some individual will show severity, or some individual will not show the severity. This is much more complex. This is depends on the genetics of the host. So overall, it depends on the host as well as the the infection dose. Okay, so all these things basically contribute to the disease outcome. That's what I can say. Am I clear to?
Yeah. Thank you so much. And another question from Dr. Leena. I guess you already explained. How can you differentiate symptoms of various RNA viruses that specifically cause respiratory associated symptoms?
Well,
Explain it.
we cannot, based on the symptom, it is very hard to differentiate different kinds of flu and cold. Basically, we need a test, a molecular test. This could be now you all are aware that there is RT-PCR or RNA. We can find out the RNA, and then we can say that, okay, individual is having a rhinovirus infection or influenza virus infection or SARS-CoV-2. We cannot distinguish based on the symptom only. The gold standard will be the find out the virus. Okay.
Oh, thank you, sir.
Yeah.
Another question?
Can we speak anything?
Any other question? One question is also there.
Yes, there is another question
Vaccine.
from Pranjal Yadav.
Pranjal Yadav. Yeah, sir. Yeah, Munil sir. Yeah, please.
Thank you, sir. There is another question from Pranjal Yadav, who says that I have heard that influenza virus vaccine causes seizures in young age of children. So is this true? He wants to know.
Yeah, I will say that I will give the diplomatic answer. I am not clinician. Well, all vaccine or any drug or vaccine, they do have some side effect, and that depends on the, again, person to person. Some people develop some kind of hypersensitivity. Some people may develop some more severe symptom. So this could be a possible, but this is not the generalized thing. Yeah, I will say that this is not the generalized thing. This can be possible in some individual. Yeah.
So there is another question from Dr. Shalini, and she wants to know that if I have influenza, does it protect me from COVID-19 also?
No, it will not, because both has a different antigenic determinants, so it will not protect from the COVID.
Another
One
question
question
from
I want to-
Arun Kumar. Arun Kumar, there is another question.
Okay.
How transmit of H3N2 influenza virus?
How?
The transmission of the H3N2 influenza virus.
Okay. So basically, they probably you might heard when there is a threat of influenza virus epidemic, or right now we did not see the pandemic. At that time, basically the government order that you should kill all this chicken. Okay, and at that time, chicken price goes very down, right? And it's a very big economic loss to the farmers or those people who rear this. So basically, this influenza virus is initiated from these birds, okay, aquatic bird as well as this chicken, and then this can hook to the animal, and or it can directly come to the human. So either way, this virus can be transmitted. Yeah, there could be a multiple way. It's not only one standard way. It can directly come, or it can come through the another. That's why probably you might heard that we call it as a zoonotic virus. They come from animal to the human, or zoonosis, the infection process.
Thank you so much, sir.
Uh-huh.
So I guess there is no more questions.
So I want to ask
thank
one
you.
question.
Yes,
Yeah,
yes, please.
So thank you for your nice talk. It was very informative and also very animated also. So I want to know one reason. Can you tell us some of the mechanisms by which favor large antigenic drifts in viruses?
Large antigenic drift.
Yes, means what
Okay.
favors
Yeah.
so much changes in their genome?
Yeah. So large antigenic drift, when you say, it means you remember that this influenza virus genome is segmented. Okay. So if the host, for example, let's take the example of duck. Okay. So duck, there could be duck and one more bird is there. Probably you are very well aware, or you might heard that bats, bat, chamkadar, right? So they are harboring all these fatal viruses. Okay. So these viruses, when one animal, one say duck or bat or chicken will be infected by multiple kind of influenza virus, and they can harbor. Reason is that they don't uh, particularly duck, they don't show any symptom, they are remain healthy. Okay. So they can harbor this virus, and when these viruses will be replicate, then they will exchange the segment of genome. Okay. And during this exchange of this genome, they can make a big changes. Okay. And in that way, they can have a different, very big or generation of more dangerous viruses.
Okay,
Is
sir.
it clear?
Thank you.
Yeah, thank you.
Yes.
Hello.
So in with regard to treatment, I also want to know whether if we increase the expression of MHC1 molecules on cells, so will it help in getting protected against the viruses?
So in physiological condition, you cannot do it like that. In physiological, in normal animal or normal human, that you can give a drug and that will increase the expression of MHC class 1 molecule. But yeah, in vitro condition, we can have a better control. We can do it. Yeah. But for that, you need a all series of, it's a not only MHC1 molecule, you need a antigen specific T, cytotoxic T cells and all those things, right? So those, putting all this thing together is not so easy, even
Yes.
in vitro condition also.
It's possible in vitro, but in vivo it's difficult.
Yeah.
Thank you so much, sir.
Yeah.
Sir, Munin sir, over to you for the vote of thanks.
Just a minute.
Ah.
Professor Himanshu ji, can you open up the camera, sir?
Yeah, sure.
Ah,
Actually, I
yes,
thought
you
there
can,
is.
No,
you can
nice to
stop
see you.
sharing and then we can see.
Just I like to convey 2 and 3 messages to you. One, as per discussion with Professor Prasad, they had gone to a selection board meeting now. So there's a real deep sense of appreciation for the way you have presented. And it was really very good, and they feel a special honor to invite you at our campus, because the last moment we have a series of one of the best lectures that has to be delivered to students in the big D block auditorium to inspire about science. The last time Dr. Sanjay Singh, the mRNA man of India, the mRNA vaccine, Genova Pharma has came over here, and he has a very mind-blowing session to make the people more curious about the vaccine technology.
Yeah.
So now from my side, then please, whenever you are in Delhi, please let us know. We will fetch you.
Yeah,
Okay.
sure.
Because you don't want to disturb your protocol that you exclusively come from here and then go back into your lab, because there are 14 researchers with whom you are working. And now from my end, that it's a really mind-blowing and a creative presentation. The way you have presented, it has taken us 30 years back in our own age of a timeline by mixing up the Tom Jerry with the Arnold Schwarzenegger.
Actually, I made it for common people. That's why if you notice that I don't put too much scientific data, those are very much complex.
Exactly.
So I wanted to explain the things in a much more simpler way to non-specialist people.
Exactly, because it's important at how we can sell science.
Yeah.
That is more important, because people are very scary to enter into the biotechnology and our technology. See the keywords like CRISPR-Cas, AI, fusion, and we used to talk about the microRNA today, and that was very beautifully made us understand. Even sometimes we also got confused at what is microRNA, how deeply it is playing a role when it is. There is a one dialogue in your movie type of presentation that cutting off mRNA and missing off a translation. That means the only one thing that is going to change anti-drifts, and you change it from meat to meat, the spellings of new to new. And that is really, really creative pedagogy. And the way you are doing science, and I can say that it is just like a heartfelt presentation for scientists and the people who are curious about science. And you made Out of the protocol, I can say कि 250 लोगों को आपने दिल थाम के पकड़ के रखा हुआ था. ये इतना easy नहीं होता, ठीक है? And we like to meet you in Bhopal, but whenever you are in Delhi, please let us know in advance. We will make
Yes.
that you will come and present to the whole Amity arena, and people have a more curious questions, and we are also helpful to our delegates for keeping alive this session by asking their questions. And as a COVID survivor, I can say the way you are expressing it out, it is making a really sense that what is happening, which is not even understandable by us, and even we can perceive it later on with our consciousness that was what was going because of the viruses in India, in our own body. And yes, it's an era of vaccine technology. So hats off to you, and please carry on this good work.
Sure, sure.
And thank you.
Thank
you,
you.
sir.
Thank you. I'm quitting the meeting.
You're just for a minute, we will put up a vote of thanks and then we can.
Okay.
Bye.
Okay.
Dr. Rawali, please.
Okay. Now I would like to extend a very hearty thanks to Professor Himanshu Kumar. We are grateful to you, sir, that you took the time out and you accepted our invitation to be a part of this PDP. And the session was very informative. It was very clear. We saw very good animations, very good slides. So it was clear to students, researchers, and all the participants. So thank you, sir, again. So I also
Thank you
thank
so
all
much.
the faculty members, research scholars, students, and other
Dr. Rawali,
participants who are attending
hello.
this event.
Just a
Yes.
minute. Just one request to Professor Himanshu. Can you give us 2 or 3 areas of life science sector which are required in next 5 years by your own expertise, because you are working with immunology, vaccines, genetic engineering. So what type
Okay.
of the manpower you are expecting to come out from the master's and the bachelor's programs of biotechnology?
Okay, so the era is changing quite rapidly, and uh, since we are in India, so we can exploit the. Now we should also have a very important hand in. You know that in the databases is flooded with the data. All rich laboratory, they do single RNA-seq and all those things, and then they dump it. Now it's a like a dictionary. Okay, so all those databases is a kind of dictionary. So we need to interrogate this dictionary by very good expertise, right? And expertise means those computational guys. Okay, it's not that era that okay, you do some leave and something and then you can make a big discovery. So it's already there. One has to interrogate, and after interrogation, you need to uh, validate it, and then you can take it to the clinic also. So, for example, if you see my website, you will find out I have shifted my major focus towards a lot of this big data analysis, and we have published one very big paper in Journal of Medical Virology. Okay. So this is all the I myself I started learning this artificial intelligence, machine learning. So now my major focus is on this thing. I have all capabilities to prove it or validate it in the cell, but for that I need a candidate molecules, and that candidate molecules can be digged out by mining this data with this modern tools, right? Modern tools, when I say it's more about the ML and AI. And in fact, if you see those paper, we have applied this ML and AI, and we kind of predicted some severity biomarkers for SARS-CoV-2. It's a lot of work. So the journal which I'm saying, the Journal of Medical Virology. So now we need a both hand. We need expertise in computational thing, as well as we should be excellent in the experimental thing. And when we will bring these two things together, we can make a miracle.
Any 2 skills for the wet lab? Any 2 skills that you can suggest for life science sector?
2 skills. So one should be this CRISPR-Cas technology is
absolutely
one of the very important thing. And now I will thank, not thank, I will say that, okay, due to SARS-CoV-2, the mRNA things are coming in picture. Yes. So now this I can, we have discovered the microRNA. now it is easy to take it forward. Okay, so this mRNA thing is also, this technology is also very important. Delivery and all those things.
Delivery, mRNA delivery systems. Thank you, sir. Thank you. And
Yeah.
that's a key coming
Yeah,
out
yeah.
of it. Thank you.
Thank you, sir. Thank you.
Thank you.
Dr.
So
Rawali,
I'm
you can
quit-
carry on, please.
Yeah, I'm quitting.
Yes, Professor.
Yes, thank you, sir. Thank you once again.
Thank you. Thank you.
Now,
Have a great day.
just an announcement for the participants that in the session 2, we will have a lecture by Mr. Gautam Bhattacharya, who is the CEO of Life Science Sector Skill Development Council. So the session will remain connected. So you can rejoin back at 2:20 because the lecture will start at 2:30. And kindly note that you have to attend all the sessions to get your e-certificate. And you can join back in the same link. Thank you. So we can now leave this session, and everybody can come back at 2:30.
Hello. Sir, can you hear me?
Yes, Dr. Prasad.
Yes, please. Sir, please go ahead.
Yes, thank you, Dr. Prasad. And on behalf of our Honorable Chancellor, Dr. Asim Chauhan, and on my personal behalf also, on behalf of entire Amity University, Gurgaon, I extend a very warm welcome to you to this 5 days professional development program on innovations, opportunities, and challenges in life science sector skilling and research. Dr. Prasad, I think it is a great idea to have this particular topic at this point of time where everyone is talking about skilling and imbibing skilling in education. And we all are, well aware that the kind of emphasis the government has putting on skilling right from last, I think, 5-6 years. I've seen that separate ministry was created, and a lot of, apart from the National Skill Development Councils, there are several sector specific skills councils being formulated. And we at Amity also visualize the same thing, and on the same line, and keeping in mind the thrust which the government is putting, and keeping also in mind the kind of future we are also visualizing. We have established Amity Skill Institute at the university in year 2014. Through this school, we have undertaken number of initiatives in this particular area, right, starting from having the collaborations with various skill sector council, including life sciences skill sector council, what we had, and we had launched a very good program in quality by our Amity Institute of Biotechnology, and similar kind of initiative we have also taken in other institution with other skill sector council, whether it is apparel skill sector council or it is any other skill sector council. So in nutshell, what I would like to pinpoint here, that the Amity University Gurgaon has given a new emphasis on skilling, and the new curriculum framework which we are introducing from academic session 23-24 onward. We have also created a separate basket for the skill related courses, which may be domain specific courses or also maybe the general courses. So again, keeping in mind the kind of thrust what we are giving at university, we are looking forward to strengthen our association with skill sector council, and especially when we look at our life science school, our school of biotechnology, and the kind of courses what we are offering at Amity Institute of Biotechnology and also under Amity School of Applied Sciences. We are surely looking forward to strengthen our association with Life Science Sector Skill Development Council, and we are open to explore the kind of new areas where we can collaborate and can impart the kind of necessary skills which are required by the graduate. And again, we found that it is one side we are talking about skilling, on another side we are talking about the academic curriculum, and these it is important at this point of time that we should combine these two things. And also, we should take your input to, make changes or revision in the curriculum itself also, so that we can have a maximum component of skilling for our students. So there are one aspect of skilling to existing students, which are undergraduate, postgraduate student. And of course, there is a second aspect, which is reskilling and upskilling of existing employee. So I think this particular platform is going to provide a great opportunity for all participants to understand the importance of skilling in life sciences area. And also, I think the kind of speaker which are going to deliver and deliberate on different topics, they are going to highlight the kind of gap which exist in industry and the kind of skill set requirement which the industry want, and accordingly the same can be imbibed and incorporated and integrated in the curriculum, so that at the end of day we should be able to produce and prepare our student as per the requirement of industry, and can minimize the skill gap which is expected and which we are delivering at the university. So surely looking forward to have a very fruitful session during next 5 years of the program. And I'm sure that at the end of the program, the participant will be equipped to understand the kind of skills set which are required by the industry from the life science graduate of today. With this few words, I once again extend a very warm welcome to Mr. Bhattacharya ji for sparing his time. I know, you were busy in the morning. There was a sudden meeting, and immediately after finishing your meeting, you have joined now, and this shows your love and affection for Amity. This shows your love and affection for student, and the kind of commitment which you have to the cause of imbibing skill among student and among the community. With these few words, thank you very much. Thank you very much, and I wish all the best to all participants.
Thank you, Dr. Madhukar. So nice of you to say these few words. I couldn't have missed this opportunity of interacting with everyone here at Amity, and once a program is scheduled, it is imperative and it is our responsibility to conduct this. So I thank you for accommodating me and the change of schedule. I couldn't help but to request you for that accommodation. So thank you so much. Thank you for your kind words.
So, thank you, Professor Sir. Now we can start the program of this afternoon. Dr. Macaulay, kindly take care from here.
Sir. A very good afternoon. Let us restart it with the formal welcome. And I have to introduce our Honorable Pro-Vice Chancellor Sir also. A very crisp, because so that we can understand who is in the what area they are working. So good afternoon, everyone, and esteemed guests, colleagues, and delegates. Greetings of the day. So on behalf of Amity University Haryana, we welcome you again at the PDP session, and that has been postponed to the afternoon. So now this is going to be considered an inaugural session because our chief guest is there. That is Shri Gautam Bhattacharya ji, CEO of Life Science Skill Sectors Development Council. And this program is already is a part of a agreement that MOU has been signed with the LSE last year during the June 2022. So we planned it like that because there is a World Youth Skill Day on 15th of July, and that was coming in the weekend. And that's why we planned it on the Monday, so that we can celebrate that day of the World Youth Skill Day. As already briefed by our Honorable Vice Chancellor Sir, that we are already in the skills from last 5 years, and there are all sectors in which the skilling is going at Amity University Haryana. So the science, in the case of the life science sector, we know that it is a range of disciplines from biotechnology to pharmaceuticals, from genetic engineering to the computational biology and the applications of the AI in CRISPR-Cas technology. So holding the platform to transform the very fabric of our society with the help of the life sciences. So this program aims to illuminate the cutting edge innovations, vast opportunities, and crucial challenges that define this dynamic industry. So I welcome all so that we can be here in the session. So just giving a very brief introduction about our Pro-Vice Chancellor Sir. So Professor Vikas Madhukar, a distinguished scholar specialized in finance and strategy, and they are a really strategist of our university in bringing out the best laurels and best academic rankings of the university. Is an exceptional academic leader in this field with a doctorate in international finance from international finance from Kurukshetra University. He currently serves as a pro vice chancellor. In addition, he holds the positions of a dean faculty of management studies and director of MIT Business School. His remarkable expertise extends to heading the MIT Innovation Incubator, a prestigious initiative supported by Ministry of Electronics and Information Technology under MIT TIE 2.0, where he spearheads entrepreneurship and startup initiatives at the university. So, Professor Madhukar's influence extends beyond academia. He also served as a resource person and trainer to the esteemed organization in the corporate world like Ericsson, Keen India, Food Corporation of India, and Cowell 2 Software. He holds as a chief analytics, showcasing his commitment to advancing knowledge and research in this field. So Professor Madhukar has been instrumental in implementing reforms in the student enrollment and has made significant contributions to various academia and administrative initiatives in key strategizing areas of the university. So we welcome our sir, and they had already given a welcome address. So now let me brief, sir, let me brief about our stalwart scientist, Professor Rajendra Prasad over here. So our director, sir, and Professor Prasad is an eminent scientist, specialization in molecular biology, holding the esteemed positions of Director, MIT Institute of Biotechnology, MIT Institute of Integrative Sciences Health, and Dean of the Faculty Science, Engineering, and Technology. So with a remarkable career of 40 years at JNU in New Delhi, he served as a professor, Dean Life Sciences, Chairperson, Molecular Medicine, and Rector. Having all the different top scholarships of the world, like you can talk about the Mercator Professorship at University of Bonn, Alexander von Humboldt Stitch Fellowship, and Royal Society Common Bursary. So all these are with the Professor Prasad, and they are with us from last 8 years in the MIT University, Haryana. So extending with this, a warm welcome to sir. So I just request my colleague, Dr. Rawali, to please introduce our chief guest today.
Thank you, Professor McKinley. I would now like to introduce our keynote speaker. So Mr. Gautam Bhattacharya is the CEO of Life Science Sector Skill Development Council and is a member of its governing body. He also serves as Joint Apprenticeship Advisor, Life Sciences to the Government of India, a pharmaceutical and healthcare professional with over 27 plus years of working experience in leading multinational pharmaceutical and medical care companies in India. Mr. Gautam has worked in senior leadership positions with companies in India like LG Life Sciences, Boston Scientific, and Win Medicare. He has acquired deep domain knowledge in pharmaceuticals, biologics, medical devices, diagnostics, vaccines, and agrochemical sectors, and has successfully managed multiple business functions. He has been a core member of management committee in the organizations he has worked with and has been instrumental in setting up long-term core strategies, especially emphasizing the development of human capital by skilling and training of team members. He has received multiple trainings and awards, including high performance sales and marketing functions in US by Boston Scientific and leadership trainings in LGLS. Passionate about building up and growing new businesses, exploring new domains, and making a difference to the society at large, he is extremely keen in collaborating with teams of multiple organizations and ethnicities from across the world. So with these words, I request you, sir, to please give your talk, and we look eagerly wait to hear you.
Thank you so much, Dr. Rohit. And everybody else, thank you so much. It's a great honor and my great pleasure to be here amongst all of you to discuss a very, very relevant topic. I think there was just a mention about the World Youth Skill Day on the 15th. And I was in Kolkata with FICCI organizing, West Bengal FICCI organizing an event on the same day on skilling. And they wanted me to be there and interact with a group who's from the academic institutions, from the industry, and from even the hospital groups and other people. And we discussed about skilling amongst youth. So as you rightly said, there's a lot of emphasis now and a lot of discussion ongoing on skilling part of it. But my personal opinion is, and having come from the industry side of it into this ecosystem, in industry, we would just not stop by saying that we want to do this. So most importantly, how do you do it is the most important thing, right? What are the actionable points? So before I start, let me just quickly give you a glimpse of the reason that why we had to reschedule this event. So Ministry of Skill Development and Entrepreneurship is the apex government body who's responsible for skilling and entrepreneurship in our country. And our minister, Honorable Minister, Mr. Development Programme was there in this meeting, and he emphasized on the E part of MSDE, that is entrepreneurship part of the MSDE. So his entire talk and emphasis was that how do we develop entrepreneurship in our country, and what are the steps to be taken, and not just talk, what are the actionable steps to be taken to deliver this mandate of creating large entrepreneurship group of people in India, so that people are not seeking job, they are providing jobs. So I'll talk about it a little later during my session when we talk about the entrepreneurship part of it. Before I start anything else, let me give you a small background of what work that I've done so far. I think that will help you all to understand that when I'm saying something from the industry point of view or from the skilling point of view, I want to correlate it with the ground level work that we do on this. So you all know my name is Gautam Bhattacharya. I've joined CEO Life Sciences Sector Skill Development Council since April of this year. Prior to this, I worked with domestic, national, and international multinational companies, Indian multinationals and US multinational companies in the field of pharmaceutical, generic, and also innovative pharmaceuticals. Medical devices, diagnostics, agrochemicals, veterinary, multiple other domains, and the last working opportunity that I had was with LG Life Sciences. LG is the same LG group of companies. They have life sciences part as well. And many people, when I say this, that I work for LG Life Sciences, so they always get astonished. Really, electronic guys, they have some life sciences products, and believe you not, LG was one of the few Asian companies, life sciences companies, who's created, we call it NCE, the new chemical entities, right? And got approvals across the world. My vision is that at some point of time, I want to see so many of our large Indian pharmaceutical companies, biopharmaceutical companies, and all the people in this meeting and the students that you interact with to innovate and create such kind of new chemical entities, which we can give to the world and say, this is India's contribution to the life sciences sector. So this is a long drawn vision that I have. And honestly, I'm telling you, we can do it. And we can easily reach that stage only if we are skilled. So now let me start my session on, my topic. Dr. Prasad asked me that what topic you would like to deliberate on. And the topic that I've given was skilling and availability of skilled manpower in life sciences sector is the single most critical factor in achieving national objectives for the life sciences sector. That means I'll tell you what are the national objectives just a couple of minutes from now. But then think about it. If we have to create a goalpost, and we created a goalpost saying that we want to become a 400 to 500 billion US dollar industry by 2048. So this is, I'm talking about just pharmaceuticals. So this is the size that we are having a vision that we can create. How do you do it? without skilled manpower, without having the adequately skilled manpower. Yeah? We all call ourselves skilled, but adequately skilled manpower. Without that, such talks and such vision will not be possible. And that's what my talk is all about today. I hope you all gain something out of it. And if you have a lot of questions, I'll be very, very happy. And then I'll feel that my talk is effective. Please don't agree to everything. If you have questions, you can just write in, or you can, tell me through Dr. Singh. Dr. Singh, I think that is fine. We will have at the end of the session some question answer sessions, correct?
Yes, sir, definitely. At the end of the session, people are going to drop their questions in the chat box, and then we'll discuss over.
Okay.
Sure, sir.
Yeah.
Please.
So,
Thank you.
So in line with the PDP theme that you are running of innovations, opportunities, and challenges in life sciences sector, skilling and research, I would like to start off with a real life story. Yeah, I know this is a post lunch session, so we should have some, blood circulation to happen. Everybody's sitting and listening, so please listen to this small real life story that I've faced. So possibly this real life story some of you must have heard, because this is in the management parlance that this is talked about in multiple places. But this is very, very relevant to our session today. So pardon me for those who's heard this and for the repetition, but I will tell you that there was an organization, a growing pharmaceutical organization in our country, and they were incepted doing small work in one state to the multiple state operations, and then from 5 crores to 10 crores to 30 crores to 50 crores. And when organizations, they hit the right pace and the right mix of products, and they know how to deliver these products to the customer, and organizations start to grow. A lot of work, a lot of hiring happens. A lot of hiring happens in such kind of organization. And these organizations, when they start to grow, they would possibly grow at a higher pace than the industry also. They need a lot of talent, and those talents are hired from left, right, and center, right, in all departments. So those talents are hired for what? To quickly come into the system and to start creating strategies of the next level of growth. So if they are lucky, they get good people to devise the right kind of strategy. And as it happens in such organization, people also get quick promotions. People get bigger roles and responsibility. And you have a mix of people at a most important position, making strategies, taking decisions for those companies. Such organizations are generally also stretched for finances. That means, although they are growing very fast and earning money, but with the appetite for expansion, sometimes they are stretched for finances, and therefore, sometimes the manpower training and skilling is compromised, because the board of directors are always look at the financials and the P&L very, very critically. So what happens one day is the HR head of this organization is extremely worried. Instead of being very happy that they are growing at such a large pace, he's a little worried, and he's actually not very happy. And he's worried because he knows that the competency of all his people at the important position is not up to the standards of what his own expectation is. And the bigger risk is that they are at a decision-making positions. That means, think about a CFO, or think about a person as a marketing head or R&D head or whoever it is, right? They are at a very, very important position for the organization, and then the HR head is feeling that maybe the competency is not very high. So what do we do? So HR head thinks hard and decides that the key people or at such position should be skilled, upskilled, reskilled, and trained well. So he walks into the CEO's room. Now he can't have just that decision all by himself, and he'll have to take a approval from CEO. And he walks into the CEO's room with a firm plan and the training calendar and the budget sheet for asking for approval from the CEO. So CEO looks up the plan, and while looking at the allocated budget and the challenges the HR head is proposing, he's challenging back the HR head. He's saying it's such a huge amount of budget you've allocated in skilling and reskilling and upskilling these guys who are already stalwarts in their field, and they've been hired at important positions and they're doing fine. We are growing. What are you saying? This is not needed at all. What if we skill them and they all leave? Because we are a growing organization, they can go to a bigger organization. And it is a very big concern that we invest so much of money and we train them, we skill them, and they leave the organization. All the investments that we will do from the organization will go waste. And hence, I will not approve it. So HR head is now looking at the CEO in his eyes, and very assertively he puts forward a point. And I want all of you to listen to it carefully, that what he's saying. He's saying that, sir, my worry is not that these people are skilled and they will leave. My worry is if these people are not skilled and they stay in the organization, what will happen? How will we grow? So now think about his observation that in our country, and the company is just an example, and this can be extrapolated and it can be made into a smaller unit. And the same example goes that amongst us, amongst whatever we are working in, if we don't have skilled people that we are working with, an unskilled person in a lab will ruin the entire effort of a product's journey. Do you know why do we get so many GMP warnings or so many US FDA warnings amongst the manufacturing sites of bigger organizations and such large organizations? It's possibly because somebody down there is not skilled enough on the GMP, good manufacturing practices. Somebody down there has not trained enough or skilled enough on the good laboratory practices. Somebody who's working in those environments is not skilled enough for good documentary practices, so GDP, good warehouse practices. This, when we reach there, when we have large number of people in the organization, such important positions, everybody is skilled, that's when we will receive or when we'll achieve excellence in those organizations. So this is the importance of skilling. And this is where one organization is different from other. If you look at large successful organization, and there was a survey which was done by one of the top 4 consulting organizations, that what is that one critical difference between various organizations or countries for that matter, who are right up there and the most successful, and somebody who's not there? And that answer is skilling, and how skilled the manpower is. If you look at the Google, you will possibly find one document which maps all the countries in the world in terms of various parameters, including skilling. So possibly we are in late 50s. Switzerland is number 1. We all vouch by Swiss chocolates and Swiss watches and the engineering might that they have. And that has not happened just like that. Not that they're born with some kind of special talent that they would make such amazing products. But they have integrated skilling into all facets of life, right from the primary education onwards. There's so much of vocational education that is delivered to all the students, and so much of skilling that happens over a period of time that when the student comes out and joins the industry, skilled. I have worked in a organization which was of Korean origin, LG. And please believe me if I am saying this, that I met an intern from Korea. So he wanted to come and do internship in India. And he wanted to do that in LG Life Sciences because we have been doing some good work and he wanted to learn that. So I had an opportunity to induct 2 interns, one from a very reputed pharmaceutical institution, a state institution, very reputed. I will not name that. And another is this guy whose name is Mr. Lee. Last name, I'll not say the full name. Last name is Mr. Lee, and he's come from Korea. When we looked at both the individuals, the background is same, almost. This Korean person could do everything possible in Excel, in Microsoft, any MS Office modules, even the project management, and his thinking of how he's breaking down the given task of the assignment into multiple projects, and then how is he planning the whole thing. And on the other hand, our national university person is not at that level. That's the importance of skilling. That's where we realized that, and we asked him that, where did you learn all these things? And he said, in the schools. And during his college days, he could learn that. And that's where we'll have to do a lot of work. So, I'll just go into the session a little more deeper into this. So from the example that I just quoted to you about a HR manager talking to the CEO, the moral is skilled manpower is an asset and single most critical and differentiating factor to decide the success or failure of any organization. And we as educators, I'm assuming that all those who are present here in this meeting are mostly educators, and we have responsibility of actually telling all the students the importance of skilling and what is skilling. It's not just, I am skilled because I've educated myself. There is a difference between being educated and being skilled. And I'm sure that you all know about this, so I'll not deliberate on that part. There's one more thought that I would want to run with you all, and then I will go on with my session. And please tell me if my thought process is right or wrong, okay? Still, when we talk about a journey of a student, let's look at a journey of a student, possibly in semi-urban or urban city with reasonable resources for education. After the class 10, still the choices are whether the student will pursue science, which is either a maths or a bio or a combination of maths, bio, engineering or medical, as we call it, commerce or arts or humanities, and some minuscule any other part. A math student would essentially appear for the engineering exams, and the biology students would essentially appear for the MBBS exams, correct? Commerce students would think about career in CA, and art students in humanities. You will have to tell me what they will do, yeah? Now, the success rate of these exams are in single digits. You all know, we all know that. And that leaves us with a huge proportion of students who have not qualified those exams, the big exams, and they are now looking for what choices they have in their life. This is where our role comes into play. How we will bring them into the mainstream. And you can comment on this later, but I have talked to about hundreds of students, and I've asked a very common question: what would you like to do? 90% of the students would like to do a computer science course. They don't know whether computer science will work out for them or not, but they would want to do a computer science course because of what? Because they'd possibly get a good job. That's it. computer science course a degree on computer science will get them a good job. This is where we have reached. And this is where I think it gets a little more scary, that if we continue like this, then we'll lose out a very critical advantage that our country has, which is the demographic advantage vis-a-vis all other countries that we compete with. We all know that we compete with all the developing nations, for the manpower and the skilled manpower that we have, and the educated manpower that we are, not called skilled manpower, educated manpower that we have. But then that thing is changing because of what is the final outcome. That means a student is only looking at possibility of getting a job after doing this entire education. It is good, but government is talking about innovation. Government is talking about entrepreneurship. Government is talking about a strengthening life sciences sector. If this is the situation, then how do we attract people in life sciences sector? I think we can attract people in life sciences sector, any other sector for that matter, by doing only one thing, is to offer them skilling in such a way that they get started with employment, or they get started with doing worthwhile things with the skills that they have acquired, whether it is entrepreneurship, whether as a research, whether they join an organization, anything that this acquired skill is helping them to further their life or further their career. I'm sure you all resonate with this thought, and please let me know how do you feel when I'm saying all these things, because this session is all about talking to mature set of people who would understand what I'm trying to say. At this stage, I would like to introduce to you a very, very path-breaking, path-breaking intervention by the government with introduction of NEP 2020, National Education Policy. You all possibly have heard and read about NEP 2020. I will not stop by explaining what is NEP, because we'll discuss a little bit about NEP later in the session. But why do I call it path breaking? When I was studying, possibly I never had a chance of having a credits for the studies that I've done, and now it is there. So one is National Credit Framework. That component is very, very critical for anybody who's taking or doing any education and getting credit out of it, including vocational education, including skilling. So if I'm doing skilling, if I'm doing a vocational education, I'll get credit out of it, and that will add to my education credits and credit bank. And second is the easy entry and exit. That means if I am a science graduate or I'm pursuing science and I want to have, I have a very strong interest in something which is arts, and I can pursue both, and I can have easy entry and exit from the courses. I think that is fabulous. Today, that will give a lot of flexibility to the students. Somebody who's not very excited about a very structured set of curriculum that is given to him, take it or leave it kind of situation, will find it interesting that there is a flexibility of the kind of education that student would want to adopt and want to pursue as per the interest of the student and as per the intellect, or not only intellect but interest of the student. So that in my mind is very good. And we as an educators, we as people who understands this, I think we have a responsibility of counseling our students and helping them in making a better educated and skilled individual out of that person, and not only wanting to have students with very fixed frame of education, who interestingly would go to the industry, and industry would say you are not skilled enough. So, in this role as CEO Life Sciences Sector Skill Council, I get to interact with a lot of academic institutions and also a lot of industrial organization, as in the companies. You'll be surprised that when I talk to the companies, what would a company say? The HR person of the company say, Gautam, I don't get enough skilled individuals from the academic institutions who are ready to kind of come in and start to work. So I have to spend time, 2 months, 3 months, 6 months, 1 year to skill that person and then get that person inducted into my organization. On the contrary, when I talk to the academic institutions, so they would say, with all the curriculum and the vocational education, everything given to the students, they don't find jobs. Now think about it, that supply side of it is academic institutions. They are saying their students are not finding enough jobs. And on the demand side of it, there are companies and industries, and they are claiming that they are not finding enough skilled manpower to induct them immediately and start running. There's something wrong. And that thing in my mind is the right amount of skilling, or the skilling for the appropriate skilling. It's not skilling whatever you want. It's skilling what is required by the industry. That brings us to a point that, do we know what industry is actually looking for? So we as a sector skill council, have a responsibility, all sector skill council for that matter, that we'll have to map the entire sector in terms of what are the most relevant skill requirements, or where is the skill gap, and then devise all our courses, curriculums, and qualifications to be provided to the students and the working professionals to skill themselves. And that is the key role of sector skill council, as in to find out skill gap. So I'll tell you about this. I have a small section on this that I want to discuss about. But if I forget, Dr. Singh, please remind me that I'll have to inform everybody about the skill gap study that we do. Yeah,
Definitely, sir.
Yeah. So is it okay that I take a brief pause and drink a glass of water?
No, sir. Our pleasure. Take it
Yes.
easy. It's your platform now.
Thanks. Just a
problem.
moment. Okay, so let us start with discussing about, and this entire session is about life sciences. So I want to make sure that everybody who's part of this group has some interface with the life sciences, right, Dr. Singh?
Yes, there are people from pharma, biotechnology, and or from life sciences.
Okay, great.
Big data
Now,
analysis.
okay, great, thanks. And if you are from life sciences, then you would understand what I'm trying to say. What is life sciences sector? Most often people ask, and I then introduce them by saying this, that the whole life sciences sector revolves around the living beings remaining healthy. So do you know the longest fought war in our evolution so far is between the microorganisms and the living beings? That is the longest war that we are ongoing right now. The COVID-19 situation was when the one virus mutated and wanted to finish off the living beings from this earth. Thankfully, living creatures like us, human beings, are smart enough to defeat the virus, and that's how COVID is behind us. And this has not happened for the first time, but this has happened multiple. And And this may sound gross, but this is life sciences in my opinion. Yeah, life sciences. And then when you say life sciences, then the domain becomes so wide. And why I'm saying this to a very, very informed group of people here is because when we sometime talk about life sciences, we get confined to only few things that we know. You know, please enrich your students for us to innovate, for us to have scientists, for us to have R&D guys, and for us to have new possibilities in life sciences. People should broaden their mindset, and people should think far beyond what we are doing right now. How, and think about it from the ideation. When somebody asks me, what is your skill council's domain, right? So life sciences domain is so vast that it starts from the ideation. That means how should people ideate of having certain products or certain intervention to do what? To manage disease and to keep living beings healthy. And just think deeper and you will see that how many footprints it touches, how many sectors, subsector touches, and you will realize that this then starts from not ideation to the research part of this, all the products, to the clinical part of the product, the validation part of the product, to the manufacturing side of it, to the quality side of it. And from quality, it goes into the warehousing, the transportation side of it, to the sales and marketing side. And these days, one of the most important thing that is ongoing right now in our country is pharmacovigilance side. That means what are we consuming as a medicine? What is the effect of that medicine in human beings after the approvals? Believe me, I think if you look at the other countries, the developed countries, they have a robust pharmacovigilance part, and we still have a lot to catch up with. Funnily, you know, this is very small things that I said about life sciences. I was having a discussion with an advocate. So one advocate, he contacted me and said, Gautam, I want to discuss something with you. That fellow is coming from US. He wanted to talk to me about skilling in the legal, medical legal cases. We have advocates in hundreds. And you all know how do we become advocates and where we take law degrees, right? And what kind of personalities, your jolly LLBs would possibly give you a glimpse of the kind of advocates that we're talking about. It's not only that, but just on the fun side of it, I'm saying. But have you ever heard some medical legal cases ever in our country? And why? It is not there. To what proportion it is there in the developed countries? This is coming, guys. This segment would come. Have we ever talked to any of the students to say that they can look at this as one viable career options? I do not know it is right or wrong. I do not know it will make a right career choice or not. But I think just to expand the horizon of our thought process, you know, this one domain can also open up multiple subdomains. And I told the lawyer friend of mine that if we have enough students who are interested to learn this, why not? Now, this is just one example. There can be multiple things that we can think about. And I'm specifically interested for something which can help in entrepreneurship, something which can bring in a quotient of innovation into the students. You know, habitually, we are all little laid back. हो जाएगा यार, जल्दी क्या है? मतलब कर लेंगे, ठीक है? I see a lack of sense of urgency in most of the people that I talk to in terms of let us do it right now, as in let's complete it by yesterday. Same thing goes with skilling. And this is where we make a decision to say, we will do it sometime later. And that's when we miss our best chance of skilling ourselves, best chance of, upskilling ourselves. I think we are about, how many, 50 of us here? Is that right?
137.
137. Oh, now hand on heart, how many of you always thought that you want to educate yourself or skill yourself in something and you are still postponing that decision? Yeah. So how different are our students and what are we supposed to say to them? I think we have reached a stage where there is an urgency of skilling and upskilling ourselves, because if we don't do this, world is moving extremely fast, and I'm 100% sure that what we are today, 1 year, 2 year down the line, 3 year, 5 year down the line, will be completely different. The way we do our work, the way we educate ourselves, the way we conduct our business, and the way we do general things will be completely different. One event of COVID changed the way we communicate. Today, I'm sitting here in my office and communicating with 137 of you seamlessly. And this never happened a couple of years back. This happened because of one event in our life. But that had triggered something in all of us and into the ecosystem, wherein ecosystem realized that there can be efficiency and there can be efficient way of delivering things, even not being physically there. So what if there is an adoption of this entire thing into our education, into the way we skill ourselves and prepare ourselves? Thankfully, like the sector skill councils are supposed to also create futuristic job roles. One of the futuristic job roles that we got approval for from National Council of Vocational Education and Training is on bioinformatics. That I will talk to Dr. Rajendra Prasad later. But we are also preparing ourselves. But all of us who are into this session, we have a joint responsibility to understand what is coming in as in the future, and how prepared are we? How much are we helping our students to envision what is coming in and preparing them to embrace such opportunities which will present to them in near future? This is our responsibility, and all of us, not only any government organization, but I think the majority of responsibility is on all the teachers. So common thing, when I was a student, I would attend a teacher taking up a class which is practical, which is giving me hopes, which is giving me a vision, and which is not only telling me what is written in the books, but how that can be implemented in real life. I'm sure all of you must be doing that. And our responsibility of doing this is all the more critical right now, because everything is available online. And those students would say that I would rather study this online unless there is something practical and deliverable which I can take away from the class that I'm attending in my classroom. I will not even go to the classroom. So the challenge is also to bring in these students into the classroom by actually engaging them with real life skilling. So in our council, we have started to think very, very critically that the curriculums that we are making, the qualifications that we are making, how relevant it is for the student to embrace it to take up that skilling and become skilled person. Trust me, if it is not relevant, nobody would take up our qualification, nobody would come and skill themselves. So it is our responsibility to provide that. So in the council, we have started to think about if we can have some micro credentials, some pockets of education, pockets of skill. That means if you are wanting to have, if you want to invest about, say, 5 days, if you want to invest about 7 days into doing a course, can you just finish it within those days? And can you get relevant skill to get started and then move into the next bigger level of skilling initiative? So lot to come actually. I get moved away from the main line of my talk to some side lanes. So I give this liberty to Dr. Singh to bring me back always to the mainstream, right?
No, sir. That's absolute, because it's a realistic world. We have to be dynamically changed. And the
Yeah.
world of the AI, chat GTP, everybody is going out of Excel now.
Correct.
With a single thing, every data has been already imported. No man is required to fill the data now. So for your information, I just like to update that along with the MIT Institute of Biotechnology, we are running an MIT Institute of Integrative Sciences and Health from now 7 years. And there is a big data analysis center in which the computational biology people and experts are there. And they have a lot of, I think, nearby 5 to 6 crore of funding projects, which are working on the data sets of the cancer and other biomarkers.
Excellent.
We have already have a set. So we can even think about that when you're talking about the 5 day, 7 day course on the computational biology and AI. So we are open to it.
Excellent.
I'd like to make you visit and see our center that also.
Sure. Absolutely. Okay, so thanks very much for Dr. Singh for this information. And this is so exciting. Actually, at some point of time, I'll also discuss about, that's my favorite subject. Where are we heading to? What is that 5-year hence that we can just see and feel, and we can almost touch and see that in front of us there are a lot of things changing. Are we aligning ourselves or not to that? And that I'll come back later. First, let us tackle the big elephant in the room: pharmaceutical sector, biopharmaceutical sector, medical device sector. These are real life sector. We are consuming pharmaceuticals, medical devices, diagnostics on an everyday basis. These are massive, large sectors. And trust me, if our students do not find themselves placed in participating in these sectors, it is a shame on us. And we should introspect that, are we providing the right kind of education to them or not? Why I say this? Because please listen to the statistics. You all know possibly, and this is out in the Google and everywhere, that India is the 3rd largest pharmaceutical market in the world by volume, not by value. We are the 14th largest, 13th or 14th largest by value. That means per unit cost of our pharmaceutical products in India is low as compared to the other countries, and thereby we are 14th in terms of value, 13th or 14th, but 3rd in terms of volume. We are called the pharmacy capital of the world. We supply to almost all countries. I do not know, except for the Mars, possibly we do not supply our pharmaceutical product, but all other countries are getting our products manufactured in India or elsewhere. Do you know what is the size? The size is about 55 billion US dollar right now. Where do we want to go? 2048. Let's talk about 2030, which is what we can see 7 years down the line. Could possibly be around 150 plus billion US dollar that we are envisaging. 2048, we want to touch about 500 billion. Look at the expansion. So and I was talking to one of the policymakers, and I said that, sir, such grand vision of taking the sector from 50 billion to 500 billion. It's a 10-fold jump in next so many years that we are talking about. How will you do it? What is the key thing that is required? the key thing is our students right now, skilled manpower, and the integration of information technology, the IT part of it. When the IT revolution came in, pharmaceutical and healthcare sector thought that, nah, this is not something that would come in and get adopted so easily within us. But sorry, but you have to embrace IT now in a big way. That's the future. But then before going to the IT part of it, I want to step back and bring you back to the large sector that we are catering to, and we are connected to, and we have a chance and possibility of contributing to, is life sciences sector. And this life sciences sector is massive. So my request to all of you, all of us who are present here is, and I repeat this in every forum, we all talk about IT. We all talk about opportunities in IT. We all talk about large paychecks that IT people get. Life sciences can become one of the largest employer, recession-free employment, and we also have a chance to contribute to the society, to whatever. So anybody with any kind of mindset who would want to either get a steady job, good life, professional life, innovation challenges, making new products, acquiring new skills in any domain, life sciences is one segment and sector. So I see a lot of students making a decision of not wanting to be part of it. Through all of us, our concentrated effort should be that how do we generate enough interest in the life sciences segment amongst the students. For that, what is the sector skill doing? I'll tell you. So with the NEP 2020, our initiatives have even gone to the schools, and many sector skill councils have started to work with the school. We have also started. So at the 6th to 8th standard, there's an intervention on introducing the students on the life sciences part. And then 9th to 12th students would possibly get a deeper glimpse into what are the possibilities in life sciences. And then I hope when they join the life sciences college education, they are interested enough, or they are, they have some deeper interest, and they feel more connected to the life sciences sector. And that's when I think a lot of interest will come into contributing to the life sciences sector, and not just remaining focused on what do we get, when do we get out of complete our education and get into the job, right? We need a little more than this. Sorry. So on the life sciences part, the goal, national goal, is to have steady supply of skilled manpower for the life sciences sector to achieve its objective of by 2024 or 25, we want to be 65 billion. And as I mentioned, 2030, we want to be 120 to 130 billion. And these are all economic survey 2021 we are talking. And significant growth is expected to come is expected in coming years from this sector. The focus purely is on skilling, and then how it will help us in making our sector more innovative. I'll just possibly stay here in this topic for another couple of minutes, and before I move into the next one. If I ask anybody, international company, for example, to tell me about what do they think about Indian life science sector, ask yourself, and just step back as a person who would want to ask that, what is the key characteristic of Indian life science sector? We would say that pharmaceuticals, biopharmaceuticals, diagnostics, medical device, everything is generic. What do I mean by generic is somebody invented this molecule elsewhere in the world. We adopted this molecule in our country. We made incremental changes in those molecules. That means atenolol as in a high blood pressure product. We added amlodipine to it, another blood pressure product, and we made it a combination product. That's the incremental thing that we have done. Also, a product which was to be given 3 times a day, we reverse engineered and we looked at what incremental innovation we can do on this, and we make it sustained release, one time. But it's not enough. When we talk about innovation, we talk about a new chemical entity that is to be developed. Some organizations in our country, like Zydus, I think some other Sun Pharma and some other organizations attempted to make NCE, and they succeeded also, I think Denmark also. But then where do we see them in those billion-dollar clubs? Can we motivate some of our students to take up this challenge of creating products which will change the way world treat that disease? 60% to 70% of medical devices that we use in our country are still imported. Can we skill our students in such a way that they can create some medical device all by themselves? These are few tasks to be undertaken by all of us and help our country in terms of providing them skilled manpower who would do not only incremental innovation, but phenomenal path-breaking innovations. That will start from all those students who are sitting in our class onwards. That will not happen all of a sudden that a student joins an organization and all of a sudden become an innovator or a scientist or something like overnight. This is a process which has to happen in the academic institution. And this is where my appeal to all of you is that if you want to contribute to the nation building, this is what you learn. Okay, so this is a mammoth task. This is not a very simple single day task that will happen overnight. The reason is I mentioned that when you compare yourself with the rest of the world, and that's where the whole comparison is. You find yourself pretty much long distance to travel before we claim ourselves, saying that we have reached there and we are doing a good job. Yeah. I just mentioned that the skilled manpower and the entire resource for future of our country will come from the institutions like Amity. And it is our joint responsibility that we fulfill this requirement of the country, of our society. I'll now touch upon a point wherein we talk about employability. You know, one of the very important component of whatever qualifications or whatever modules that we create for you to do skilling or for us to do skilling is employability. That means whatever we are doing, is it really helping the students to get some employment, or is it getting a certificate? it is important for us and National Council of Vocational Education and Training, this is the regulatory authorities, when we create our qualification, they insist on creating this employment part of it in the skilling. I have a different thought on this. Remember, I was talking to you all about when I go to the industry, they say they don't get, readily skilled students to just join the organization, start working. And when I go to the university, they say that our students do not find job, and there's a gap. This is exactly what I was referring to. That how do we improve the employability part of the skilling? Please think about it. And all of us, we have to own this up in terms of our joint responsibility. That when we are educating our students, when we are talking to this group of people who are now wanting to enter into the workforce, have we given them practical training? Have we, do they know, have they touched the machines? Have they seen those machines? Have they had a chance to experiment something and add one chemical into the other and seen exactly what is happening, or at least know that what is the layout of an organization, what is the layout of a manufacturing site, how a warehouse is looking like, how tons of products are stored in the warehouse and then it is shipped right precisely by adopting certain methodologies. They will all study it in the books, but half of them would just study it for the sake of passing the exams. And that key differentiator would be that we will increase the employability of that students if we do, if we give them a glimpse of how is it that the actual factory setting is, or the manufacturing setting is, how is this gas chromatography machine looks like. For that, reach out to the industries, bring them into your campuses, into your departments, into your day-to-day life. Please have one day out of a week to bring those industry interfaces. And once they come in and they present their part of the story to the students, invite the students, ask them to invite students, some students to go into their workplace and see how do they work. And I'll give you a live example. You remember I talked to you about 2 trainees recently in past organization that I hired. When they came in, they were sitting in the conference hall, and I asked both of them, have you seen a live organization, as in a company with table chair and people sitting there with computers and some people talking on phone to other colleagues, nationally, internationally, and other things, and some boardrooms and other things? The student from Korea, he mentioned that he did, and they started off with a small company tour when they were in school, that they were taken into the company where their fathers were working or whatever, as a concerted effort of introducing themselves, introducing those students to how their parents work. From there, I think some Indian companies also do, but not as an average if I talk about. But what about the college students? If you're a pharmacy graduate, you're studying pharmacy, a B pharmacy or diploma in pharmacy, have you gone into the pharmaceutical organization and seen those machines by yourself? Not in the university labs, but into the organization. And that's where we'll have to do a very, very big effort of bridging this academia and industry collaboration. We will do our bit from a secretarial council, but any one of you who's listening to me right now, please create some opportunity, not for you, but for your students to have that interface. It changes the life of student. the way they will talk, the way they will comprehend certain things. So I'll give you an example. When I was in the industry, I was invited by one university to interact with BSc biology students. Okay, so they study those botany, zoology, chemistry, these subjects, and professor of zoology was my friend, and he said that, Gautam, can you come in and just interact and talk to our students and then tell them how is zoology spanning out in your day-to-day working. Just think about it. So, okay. So I went into the class and I talked to the zoology students and I asked them, why do you study zoology? So half of them, they study because it's their part of the curriculum. And so I asked, what is the natural relevance of it when you go and join into any company? Other than possibly understanding those science topics, nobody could answer how it will get translated into the real life day-to-day working. And that's where we are. I gave them a couple of examples to prove that they can utilize it. And then they say, why I'm saying this is because of one comment that I received. They said that the way they look at that chapter of zoology has changed. It is not mugging up those theories. It is seeing it from the practical point of view. So when you bring in those people who are in the industry, who are working in the field, into the classroom, it changes the perspective of the students who are studying that course or who's just referring to the theory part of it. So I eagerly urge all of you to please think about collaborating with the companies and the industries, to bring them into your classrooms, have a nice session with them. Half of them would possibly want to do it just without any fee also, but if that works out well, please do. And that's how I think one of the most important challenges that we have is the gap between supply and demand would possibly lessen. So bring industry people into your classroom and take your classroom into the industry. Try and do that. Make a concerted effort. And I'd eagerly appeal to all of you that if you have a possibility of doing this, you should not miss this and you should do. How are we doing, Dr. Singh, with time?
I think the time is there. There is a now really we require a discussion session with you, sir. Because you already raised those key parameters.
Okay.
Which we have to now fill the gap.
Okay.
So it is very much interesting because you have given a very good keyword that is it's a recession-free industry.
Yeah.
Exactly when we talk about life sciences, even in the case of a lockdown or some war may going on in the Ukraine, but you require drugs.
Yeah, absolutely.
And as we talk about the diabetes and obesity and the heart markers of the India is the biggest capital. So when we talk about these 4 parameters about clinicals, pharmaceuticals, diagnostics, these are the 3 key parameters. So here we are already in the biotech, big data, pharma, medical schools, and linking up. The thing is that we are lacking is to linking up. When we are talking about industry, yes, industry is okay. But as we see the GMP, GLP, and GDP protocols of the industry, only they open up when somebody land up in industry. 22 years back, I was an intern in some alpha drugs for 2 months, and I sensed that how TMP is packed and how TMP is manufactured, trimethoprim, and they were making it for the GlaxoSmithKline. So that was the thing which made us realize that industry is entirely a serious business. And when we approach and knock the door of an industry that we come for a visit, they are really very much inhibited because they don't open a gate. So there is a main role of a council is that because we are already in connect with you from last 3 years, but now in the MOU from last 1 year. So how can we make this gate pass or opening into industry, because we require the interns has to be gone by your route. When my training and placement go and knock their door, they assume that it is some mandate they have to, because when it go through a policy maker or the council, because you are reporting to the ministries. Okay. So if our 5 or 6 guys go by your route, and that makes some more different sense, because the parameters that we have discussed regarding the filling up the gap and the flexibility, yes, in NEP 2022, we are in. Media has already taken over NEP 2020, and we are redrafting our curriculums. And with the next batch that is going to come from over the next month, they are coming up with the NEP. All those micro credits and major credits are already running in our organization. But as per with the NEP, we are going to switch on with them. And regarding the, when we talk about the internships and we talk about the bridging, so there we are going to get more information regarding how can we make a bridge a gaps. You discuss about GDP, GLP, GMP, and we have all those equipment. When you talk about the GPS, HPLC, you talk about the UPLC, you talk about the flow cytometry, microscopy. So there is a lack. That is lack not because of academia. Academia is its own curriculum, industry has its own. So
True.
you as between can play a bridge role.
Yeah.
And that I already requested with Ankit a lot of times that how can you make up a bridge? Because time is the main parameter. And we talk about we know we have a very big case of white elephant dying his own death around taxi. I'm very much open in industry, okay? And we talk about the Novartis Gleevec case.
Yeah.
Everybody knows these two cases. So and now it is our, as academicians, we have a charge on our head and industry. So we have to fix, because there is a difference between a Korean intern and an Indian intern. But who will own the charge to make our intern more Korean intern?
Correct. Correct. Absolutely. And I fully echo what you're saying. Let me just quickly give you a glimpse of what can be a possible opportunity areas, and then we can discuss more on that.
Definitely.
Is an initiative of the government called apprenticeship course. Okay. It's in National Apprenticeship Portal is there. So industry is wanting to have entry-level workforce into various domains. And we are in discussion with the companies and industry associations to increase their uptake of apprentices. If our students who are wanting to kind of join industry, join as apprentices in many companies, I think that can be one important agenda that we can further in terms of industry getting students who are ready to learn, and it is on the job. That means you enter, you are taken in as apprentice, you prove yourself for 1 year or 6 months to 1 year, do well, get absorbed into the company. If you're not good enough, you need more skilling, you skill yourself, go back to the industry. That one very important agenda of the government is on the apprenticeship should be discussed more, and we should see that how do we utilize this to bridge, as what you are saying, in terms of how, under what scheme can we have these placements done in various industries. Let's discuss a little more on that. Yeah, I think we have a chance.
Yes.
So why not we open the floor for some question answer sessions, right? That's what you said.
Dr. Rawali, you can open up. As we also request you to make up a visit with your team, and you can see our institutes, facilities, and the lab facilities, and you can come know what we are going to give them on hand. That's more important.
Sure, absolutely. Sure.
Definitely. Sure. Dr. Rawali, open with the question.
Yes, thank you, sir, for your nice talk and deliberations. So one thing I wanted to ask is regarding the gap between the industry expectations and the academic institutions. So this has been a problem at all levels. I think at the school level, if we can train the students at university level, then this would address the issue. So what do you think, what major steps can be taken starting from school level to university level and even at higher education to bridge this gap? How can industry be made a part of curriculum design, which is acceptable to all?
Thank you for asking this question, Dr. Ruveri. Let's ask ourselves some questions. It's sometimes easy for us to give this to somebody else to answer for us, but let's start with a finish line first, a goal post. What do we want? We want industry people should be part of curriculum setting, or should be part of education system, our education system, or delivering of our course in certain way, industry relevant way to our students, right? This is what we want. How many attempts have we made to bring them in? I'll tell you honestly, and no offense meant for anybody, but I want to have open, fair discussion here. What happens is, when I look at the curriculum, not MIT, I'm talking about general academic institutions, and try and say that let's bring in this change. Let's change this as per the industry requirement. It has many, many ramifications, that how it is then correlating with X, Y, Z, and multiple other things. I don't have to really spell out those things, but I hope you understand what I'm trying to say. We will have to be flexible to adopt what industry needs and wants. Otherwise, just think about it. If industry has certain requirement, is it not very clear and easy way to bring in those people? Like, for example, you go to the HR department of an industry, just make a connect with, say, the India's largest pharmaceutical organization, or the best pharma company, one of the best pharma company, biotech company, and other things, and make a representation saying that we are from this university and we want to kind of have a workshop with you. Can you send your HR person here? And we would like to know that what kind of people do you hire? What skill sets do you see in those people, and what education and what exactly you are looking for? And then you note it down. Yeah. And when you are making a curriculum, you just include those things, and then go back to the same organization, say that, you had told us this to be done, we did this, and this is what our students know. And when they check with the students, they realize, oh yes, these students know about this. Is it difficult? But the difficulty is execution. The difficulty is organizing this whole thing. And no offense meant to anybody. Please don't misunderstand me. Yeah. I know it is easier said than done. I know that. Yeah. I run an organization wherein sometimes it is not very easy to be flexible and change things very quickly, because you are answerable to multiple organizations and various stakeholders, right? Sure. But let us start. My request to you is, can you send out invites to multiple organizations around you and ask their HR heads or quality heads or their manufacturing heads or their sales and marketing heads to come and take a session once every week to the students? You will see the change. You will see the difference. You don't have to change anything, but it is adoption of the best policies from them. So I think that is to be increased. No, Dr. Devan.
So we have basically two options: either to integrate these skills in the curricula, or to have an extra, say, course or training sessions for these students.
Possible, yes.
Thank you, sir. So any other question? Professor McEvely? So there is one question from our faculty, and she wants to know, Dr. Sangeeta wants to know, how many biotechnology related skilling centers we have in Delhi NCR, which we can inform the students.
I'm not sure I've understood this question very well. So you want to know that how many training centers or the training institute we have related to biotechnology? Is that what you want to know?
Yes, means how are there specific centers where students can approach and get some skilling say diploma or certificates, or how they can do these things with extra learnings?
And add on to, sir, actually there is a dire need when we are talking about the skilling, that's absolute, but recognized and a certified skilling is more important.
Yeah.
Because there are a
You're
lot of organizations where we are skilling, but
Yeah,
when
you're
they
right.
pick up the money, they come to road and there is nothing. So
Yeah.
dire need in state also for on the NCR for developing the certified biotech skilling centers.
You're right. So I fully agree. And this possibly you've raised a very important point. What is the role of a sector skill council if private organization can do all the skilling? Okay, no need. When I joined the sector skill council, the first 1 week, I asked this question: oh, why? You know, I'm a very curious person. Right from childhood, I was very curious. I wanted to know why moon is so far from Earth. So the role of sector skill council is to create standards, national occupation standards. They are the standards. If you skill the person with those standards, with those qualifications, there is a occupational standards that you have met. And once the candidate is assessed and certified, you know that this person who skilled himself or herself into this job role or this qualification has met the national standards and therefore received assessment or certification. And to your point, Dr. Wali, Dr. Singh is absolutely right. Anybody and everybody can do skilling and can do certification, but is it that standards or not is very important. I would like to answer to Dr. Sangeeta. Dr. Sangeeta, are you here? I can see you, Dr. Sangeeta Kumar. Can we have a little dialogue between us, Dr. Sangeeta?
Yes, sir. Even
Actually, she
Professor Prasad has
posted
joined.
this question.
Yes, sir.
Yeah, Sangeeta ji, can you please elaborate your question?
Sir, just we want skilling related centers at Delhi NCR.
Okay, what do you mean by skilling related centers? Because for me, most of the skilling happens in your institution. इससे बड़ा skilling कहीं नहीं होता.
Yes, we know. Is there any specific skilling related center?
जी है, बहुत सारे हैं.
हाँ,
अब आप मेरे को बताएं, BTech biotechnology or biotechnology is such vast word and world, right?
Yes, sir.
We will have to be very specific in terms of what are we looking at. So,
Sir,
why not we
biotechnology, related to biotechnology,
connect? Yeah. We
life
have.
sciences.
So let's connect after this call separately,
Right, sir.
and I'll tell you exactly what all things are possible under biotechnology for skilling into different domains.
All right, sir. Thank you, sir. Thank
And my answer is yes, we have.
Okay, sir. Thank you, sir. I just
Thank you.
wanted to know this thing.
Sure.
Thank you.
Thank you.
Gautamji, I'm here.
Sir.
And very, very informative insight. We only want to just flag out the diploma course which we started on skilling, particularly with your blessings from your council. The discussion was that you guys will help us in giving some industrial or some kind of student for this course. Because you have more connection with the industry for upskilling. So unfortunately, we have not been able to launch this course properly yet.
Okay.
Because on one side, we do not get many students from here, but also from your promise side from the industry, also we are not getting any response. So if that can be sort of accelerated, maybe we can. And one more.
Yeah.
Because of the new education policy, now we are trying to make it mandatory after mapping the course structure as a 4th year.
Okay.
As a requirement, they will do this kind of course that will be also part of skilling. So when
Okay.
they graduate, they would have already done 1 year skilling course also. This is from our
Yes.
side.
So Dr. Prasad, allow me to come over and have a word with you separately on
Yeah.
this. As you know that I joined this council in April, and I'm extremely enthusiastic to work together and to look at it once again.
Perfect. Perfect.
Let's have a discussion on this. I cannot comment on it just in without knowing deeper things into it. What did we plan? How did we plan? What were the deliverables? What have we done on that? And various other things. So मैं थोड़ा सा इस तरह का इंसान हूँ साहब, मेरे को थोड़ा माफ करिएगा, कुछ समझ में नहीं आता तो फिर मैं आंसर नहीं कर सकता।
नहीं, मैं आपको बुलाता हूँ।
जी सर,
हम लोग आपसे बात करते हैं।
जरूर इस पे करते हैं।
Mike, are you there? सर को 15 अगस्त से पहले बुलाओ।
Yes, sir.
Then we'll see.
Definitely, sir. We will
Fine,
call them with their whole delegation, sir.
Yes, sir.
ठीक है,
So-
करते हैं सर।
Definitely.
Okay. So we will make out a program with your consultation
Sure.
and we start from there very soon.
जरूर।
Thank you, sir. So nice.
Thank you.
And Dr. Rawali, you can offer the vote of thanks, sir.
Okay.
Thank you, sir.
So thank you, sir, for a very nice and informative presentation. So I would like to extend a very hearty thanks to Mr. Gautam Bhattacharya for his nice lecture. So we are grateful to you, sir, for taking your time out of your busy schedule and joining us and sharing your knowledge with us for a very important topic, that is skilling. As being skilled is not same as educated. So we now understand the importance of this. So I also thank all the faculty members, research scholars, students, other participants who have joined this PDP, and I hope they have gained a lot of new insights. So thank you all for your support, and I expect to see you again tomorrow at 11 a.m. in session 1 for a talk by Dr. Diksha Tripathi from Central University of Rajasthan, and it will be entitled Mycobacterium tuberculosis, New Insight into an Old Bug. So please join again tomorrow, and we hope to see you again. Thank you all. We will meet again tomorrow.
Namaskar, sir.
Thank you so much. Pleasure being here.
Please share the feedback. Munindra sir,
I
Please
have
share
sent the
the feed-
feedback. I have sent the feedback link in the chat section, so all of you can fill it.
Yeah, my request would be that if you have any questions, any suggestions, anything that you feel sharing a feedback with Sector Skill Council, feel free to write to us. We eagerly wait for your response, and just don't bother about what you're wanting to write. Just feel free to write a feedback. That is very important for us to have your feedback, because that's critical for us to improve our deliverables also. So thank you very much for your patient listening. It's a great honor for me to being with you all here. Thank you so much.
Thank you, sir.
Thank you.
Thank you, sir. Looking forward to
Thank
see
you.
you soon in the campus soon.
Sure. Thank you so much. I look forward to
Welcome, sir. Thank you.
Thank you.
Bye, sir. Bye.
Bye, sir. Bye. We can
Thank
close
you.
the session now.